society scholars, has made significant contributions to cancer biology. He has given part in publishing many books and has published 3 patents. He has given a contributions by attending and organizing many conferences, seminars, and workshops. Today, she is leading this session and introducing our distinguished speakers. Without further ado, please join me in welcoming our patient guest, Dr.Nalla G, to introduce our teaching. Thank you. Hailing from the city of beauty, nickname of Chandigarh, we have with us Dr.G. Chief Scientist, CSIR, Institute for Microbial Technology, INTEC. It is indeed a dream come true for any life science student to become a Chief Scientist at CSIR. Let us see what paved way for Sir's success. Sir completed his bachelor's from Madurai University and master's from Harvard University, obtained grad fellowship from University of Munich, Germany, and then further moved to INTEC to pursue his Ph.D. and then postdoctoral research from the Ohio State University, United States. As a student, he was conferred the title Best Graduate in Botany and Best Graduate of the Year in 1991 by Harvard University. He obtained travel grant to attend the International Symposium on Trends in Microbiology in Kolkata during 1995 during his master's degree. He was selected as winner of the 3rd International Research Exposition at the Ohio State University in the United States in 2009. Received the National Bioscience Award for Career Development in 2013 and took top. He was awarded the Prominent Researcher in First AMR Scoping Bespoke India by DBT and Research Council of United Kingdom. Returning back to India, since 2007, he has been serving INTEC at various capacities like Principal Scientist, Senior Principal Scientist, and currently serves as Chief Scientist. He is an expert in many domains like antimicrobial resistance, molecular microbiology, biotherapeutics, and biotechnology. Dr. Rajamohan Sir has more than 30 publications and 12 national and international patents. He has completed several government-funded research projects and received grants from ICMR, DBT, DST, CSIR, and BRAC. Sir has guided more than 20 research scholars and has been a principal investigator and also co-investigator in more than 15 government-funded research projects. He holds a very rich academic experience of over 30 years, which has created an avenue for him to be a very active member in several institutional, academic, and administrative committees. He is also a reviewer and editor for many international peer-reviewed journals. He is also serving as a grant reviewer for ICMR, CSIR, DBT, and BRAC projects, and has guided more than 50 doctoral students. With these few words, I call upon our eminent speaker to take over the session. Welcome, sir.
Okay, thank you. I am audible, right? Yeah, thank you for the nice introduction and apologies for not joining there. And it's today's session, we collaborate always science as much as possible. And today has been great occasion in terms of Louis Pasteur as well as the Science Day. Okay. So I think I will start my presentation in terms of, I'll share my slides, just see. I think I would have joined even Dr. Manikam, I could see there. So, yeah, fine. I missed My slides are visible now.
Yes, sir.
I'm just looking at and trying. Visible, right? Slides are moving, right?
Yes, sir.
Okay. Yeah, I'm just starting now. The title today is antimicrobial resistance and One Health perspectives. So these are very important two topics, like which is nothing but say, like you can always say it's a two sides of the same coin, AMR and One Health, and these are the before going into the detail of the talk. You can understand. I'm going to concentrate more on AMR molecular mechanisms and AMR gene regulations and One Health. So these specifically AMR gene regulations, I'm going to say, in terms of the research what we are doing at Institute of Microbial Technology, specifically from my lab research portions, which I'm going to share a little bit about this, some stories on that. So in the last part, I can talk about a little bit about the intake, and as well as I also would like to say one quick solutions for AMR using some WHO recommended techniques that will come. So I will start with this background. AMR doesn't need much attention, not much of introduction needed, because yesterday also you started, so people are much aware of all the AMR problems, but always a lot of statistical implications. The people are saying, AMR is a big problem, right? And if it is not contained, it is going to be the number one killer. It's going to, by 2050, they are estimating almost 10 million. It is going to cross beyond cancer. And of course, currently 1.27 million deaths are being observed from MDR, antibiotic resistant bacteria. And India also accounts for more than 58,000 newborn dies every year due to drug resistant infections. And every 2 minutes, almost 5 people are dying. So these are the some sort of very pathetic information in terms of understanding the severity of AMR. But in the discovery part, we don't have proper antibiotics now, because the antibiotic discovery is completely gone down. If you see, like in the graph, 1 second, I'll just, sorry, 1 second. One second, okay. Sorry, 1 second. I'm just putting for cursor. Somehow the pointer is coming, okay. Able to see my slides now? Hello? Please, just try to say otherwise. No, I thought probably I missed the track. Just 1 second. Just put my pointer. So, if you see the graph, the resistance is keep on increasing. We don't have antibiotics, so the antibiotic discovery pipeline is completely dry. The reason behind is all the drugs, whatever are being produced by the industry, the bacteria is becoming resistant. So the whole pipeline is very slow. So the antibiotic resistance or antimicrobial resistance largely it accounts for either bacteria, fungus, or virus or parasites. Basically, this AMR is nothing but the resistance which is developed against any microorganisms, and these are whatever the drugs what we use for treating these microorganisms become ineffective because they have developed some resistant mechanisms. So naturally, if you see these all the bacteria, because bacteria is evolved 4 billion years ago, it is known to have lot of bacteria produces antibiotics. Some bacteria, many bacteria also doesn't become always naturally resistant. So you can see resistant phenomenon naturally also, but it's not something evolved in the clinical practice. But even Alexander twice we want that use of like antibiotic, especially the misuse of antibiotics, going to have antibiotic resistant problems. So that's a warning which probably we are also having right now. Like we have the same problem of AMR. How these bacteria become resistant? Primarily, the multiple modes. The primary mechanism is one way is vertical transmission, that is under any antibiotic selection pressure. We are undergoing so it can develop resistance. So bacteria, you can easily develop any bacteria in a laboratory by adding any antibiotics, antibiotic resistance. So the other means of transfer of genetic elements will be bacteria, would be horizontal transfer mechanism, that is either transformation, transduction, or conjugation. These are the elementally we know significantly these bacteria undergoes the genetic exchange material based on these processes. And very recently, people have identified a new mechanism is called vesicle-mediated gene transfer, because most of the gram-negative bacteria produces a lot of vesicles on the membranes. Okay, these membrane vesicles also have a lot of resistant genes inside, and it can migrate to other bacteria. So these are the one of the recent mechanisms people have identified. The bacteria also undergoes gene transfer through vesicles. So besides, generally in terms, bacteria have a resistant mechanisms, either innate, that means intrinsically resistant to certain class of antibiotics, and of course bacteria have capacity to acquire always new resistant genes, be it either vertical or horizontal gene transfer mechanisms. And of course, adaptive resistance is the major mechanisms through chromosomally. All the bacteria have certain genes, and these genes play a major role in developing resistance to different type of bacteria. So if you see closely for any bacteria, the molecular mechanisms have generally in terms like outer membrane protein or called porins. These porins normally try to take the antibiotics inside the cell, and once all the mutations, any mutation occur on these porins, the drug is not able to come inside, so the outer membrane permeability is completely lost. So the drug is not able to go, the bacteria become resistant. This is the one mechanism where we can always frequently find. And other thing is bacteria have a typical efflux pumps. These pumps are the transporters which can throw efflux out the whole antibiotics. Whatever the antibiotics being used, these bacteria throws efficiently all the drugs out of these efflux pump proteins. So if these pumps are being highly expressed, that means it can throw a lot of antibiotics out, and thereby it can make the bacteria more resistant. So the other important activity, for becoming resistant, always by the beta lactams. Beta lactamases are the enzyme which you can see inherently in most of the bacteria, typically in gram-negatives, you can see. So therefore, whenever you are using antibiotic, specifically beta lactams, you can these enzymes are the one which can chew up all the antibiotics, and thereby it becomes inactive, specifically against antibiotic beta lactam. So, and other thing is if you have any aminoglycoside acid type of antibiotics. A lot of bacteria also releases aminoglycoside acid modifying enzymes. These enzymes are able to chew up all the active and aminoglycoside, and it becomes ineffective in treating the aminoglycoside antibiotics. So these are some of the elemental mechanism through which bacteria always try to cope up the resistance, that shows almost this is the prevalent mechanisms. You can see it in gram positive as well as gram negative bacteria. And besides, a lot of new mechanisms keep on coming. One is something like people always hear about the biofilm formation, because most of the bacteria have ability to form biofilms. The moment biofilm forms, your drug is not able to come inside, so thereby bacteria become resistant. So these are some of the important mechanisms you can always see. And these pumps, I said, flex pumps are the major the proteins which are found on almost all the bacteria, because these pumps are largely classified into 6 families. And any bacteria if you take, you can see not only one gene, you can see multiple genes, any E. coli, 30-40 flex pumps are there. So primarily for the resistant mechanism is mediated by these flex pumps for any bacteria. And the other mechanisms always you can say insertion sequences or transposons. Classically, if you see, transposons are the one which can called as a jumping genes. That means it can jump from plasmids or to the chromosome, or chromosome to plasmids. These are the one which are able to evolve a lot of new resistant genes in bacteria. So besides this, plasmids also have a major role, and other conjugative elements and transposons. So, and importantly, we can see gene mutations. This is specifically always if you see chlorophenicols, quinones, quinol group of antibiotics, fluoroquinone, ciprofloxacin, norfloxacin, all those things. If you bacteria become resistant specifically by mutating gyrase subunit, that is a topoisomerase enzyme, where specific region is called quinone determining region, that is quinone resistant determining region, where you can see 83 amino acid is always changed from serine to isoleucine or arginine. So you can always see this active site pocket is changed genetically in a topoisomerase gene. The bacteria become resistant to ciprofloxacin. So this is the first primary mechanism through which all the quinones become resistant. And besides, other factors also there. It's not only this mutation is the only one which can cater fluoroquinol resistance, and of course other mechanisms also involved in doing the similar fluoroquinol resistance. And the other part is you can see integrons. Integrons are the very classically you can see is called as a gene capture and expression system. This is an independent genetic mobile element, where if you have an integron in any bacteria, that is having integrons always carry a lot of resistant genes. It's not something it sits idle. If it's there, that means it is a resistant, highly resistant. It will give resistance to the host wherever it is carrying it. So broadly, if you see all the mechanisms, importantly, one has to always understand the beta-lactamases is a big group of antibiotic-resistant superfamilies, where beta-lactamases are largely classified into class A, class C, class D, and class B, based on the active sites of the enzyme. One is serine-based, or maybe the metallo-beta-lactamases. So currently, all these become very important because metallo-beta-lactamases doesn't have any specific inheritors. That's a whole problem right now we have in clinical situations where bacteria acquire NDM, that is usually beta lactamases. Then we don't have any options for any drugs are available for treatment. So that's a problem where all metal or beta lactamases carrying bacteria having NDMs or variants of NDM is a problem, because we don't have a drugs to treat besides beta lactamases, which is largely falls on the carbapenem type of antibiotics. So NDM, if you say people know about the NDM, and the important point of NDM is a lot of variants, though it is being identified in India, and a lot of variants, and it is moved in say 9 to 10 years. Almost throughout the world you can see NDM is prevalently found in all the bacteria. The carbapenem resistance is largely mediated by this NDM presence, and even people recently also identified this, also found the bacteria which is found in the Arctics also find a similar gene. So it is moving without any boundaries. The resistant genes are keep on moving. But sorry. So if you see all the transmissions, which is now always say it as a multi-layered, it's not a single gene. If it is there, the bacteria becomes problematic resistance. No, it's this is a model where now people have shown it in with this is a Russian doll model where to knit this doll, you need to have a lot of fabric. So similar in that fashion, the development of antibiotic resistance in the bacteria, it's mediated by a different set of parameters. Largely, it is driven at a genetic level. It's called a gene. If you see have a resistant genes, suppose this resistant gene sits on the integron, which I said that it is a mobile genetic elements, and it can move to irrespective of any bacteria, that means any bacterial host, gram positive, negatives, and once these integrons, it may go and integrate in a plasmid or in the chromosome of a bacteria. That means if it is on the plasmid, then again it keep on moves to the different bacterial clones. That means different bacteria will be having different plasmids, and it can also move from one bacteria to other bacteria, and ultimately it comes to the humans, and the whole process is compromised in terms of you develop high resistance once you land in hospital, or maybe you develop more resistance in the hospital. So these are the whole circuitry where you can understand how bacteria develop resistance, and these developed resistance are being transmitted from humans within bacterial milieu. It transmits very efficiently, and it also leads to potential problem. So the WHO and Indian government of India also followed the similar priority pathogens. These are clearly they classified as a critical, high and medium. These are different bacteria which requires immediate attention at a global level, because the reason behind this, we don't have any antibiotics to treat the bacteria, specifically the critical groups, which is mostly on the gram-negative sites. So with this background, I'm going to take you in a couple of more minutes, just to detail about what we are doing at IMTech in terms of understanding the antibiotic resistance, and I will give you a little bit of understanding how one can study about antibiotic resistance, and how we can find some novel gene, or because you need to identify a new gene in order to develop some new drugs. Otherwise, it's difficult to to identify drugs against a certain pathogen. So my lab is focuses on bacterial signaling and drug resistance laboratory with a major focus on two important questions. One is to understand the evolution of AMR with respect to emergence, persistence, and dissemination. The other part is just to identify what are the different signals from which can induce antibiotic resistance in bacteria, host, or environment. So largely what we do is we try to understand the local epidemiological studies in terms of how these bacteria are evolved within a locally in terms of what are different antibiotic resistant patterns, what are different resistant mechanisms are being there in different bacteria, and completely try to understand the whole biology, because without knowing the biology of any specific bacteria, you cannot develop any drugs, because whenever you are making any drugs, without understanding the bacterial metabolism or bacterial physiology, it is very difficult, because then it's going to be definitely not a success story. So we use a model organism as a escape, that is Acinetobacter baumannii, Klebsiella pneumoniae, E. coli, Clostridium difficile. We are also working on anaerobic bacteria. These are some of the model organisms which we are working to try to get the answer of specifically understanding the antibiotic resistance mechanisms, as well as getting something we can identify new antimicrobial drug targets or something like that. So today I'll be talking much about only the few short stories from Acinetobacter baumannii. Acinetobacter baumannii is a gram-negative bacteria which is strictly aerobic, and it is widely distributed in nature. You can find it in environment anywhere, and hospital also, very importantly. And importantly, this causes very serious infections, mostly in immunocompromised patients. And the beauty of this bacteria is always multi-drug resistant. So importantly, this bacteria is not only resistant, and this is have a capacity to acquire a lot of resistant genes, and it can withstand on abiotic surfaces for years and years. It is very difficult to treat these bacteria, because we don't have specific drugs. So there is much need to develop new drugs. And whatever the drugs we make, the bacteria become resistant. So we need to develop a drug which should not have a resistant problem again. So largely, these are the treatment regimes. People try to treat Acinetobacter infections through aminoglycoside fluoroquinolones, and the last resort is carbapenems or beta-lactam antibiotics. And if these set of antibiotics are always become resistant, then it is difficult to treat these bacteria. So what we do is whenever we try to start our research program, we try to understand the local epidemiology in terms of what is the resistant prevalently found in Acinetobacter baumannii strains, because we collect Acinetobacter baumannii isolates from Chandigarh, some we got from Chennai and other parts from Delhi. Routinely we keep on doing it. So what generally we try to understand is first we make an antibiogram in terms of doing a disk diffusion assays, the elementally one has to do to determine the antibiotic resistant profile for any bacteria. And once you make and you understand whatever the collection of more than 200 strains we have, and these strains are highly multi-drug resistant. And interestingly, what we found always is every time we try to understand the whole process, we land in identification of novel genes. Always you can see Bla-Indium is always you have lot of resistant genes you can see. And the important point is these resistant genes not only found in almost all the strains, and you can have a variety of resistant genes in a bacteria. So you can always have co-combination of oxacillins or cephalospore invariants. That means, the bacteria can produce different types of resistant genes at the same time. That is a little bit of a problem side. Like, these bacteria are evolved with highly resistant behavior. So not only these strains are resistant, but interestingly what we found, and many people throughout the globe also found, like whatever the biocides which we use for cleaning the surfaces, or routinely we use mouthwash like chlorhexidine, benzylcholine chloride or triclosan, and these antibiotics also, like these biocides, these bacteria become very much resistant towards these biocides. So what we found is like whatever the bacteria is not only resistant towards antibiotics but also become resistant to biocides. That is the reason why these bacteria are able to sustain on aerotic surfaces for a very long time, because irrespective of a cleaning procedures, it is not completely moved out of the environment. Secondly, we also try to always capture the colony morphotypes. This is now simultaneously, as a microbiologist, one has to keep on exploring it, because looking into the mechanisms always is tricky in terms of understanding the whole cell morphology, and try to see whether it is able to make a lot of capsules or not. So that way we keep on monitor for the different hours or different days, keep on seeing it, whether these bacteria are able to have typical characteristics in terms of persistence or emerging of new things. Because what we found is, you can see a lot of small colonies keep on emerging after many hours of incubations. These are the ones which are called as a microcolonies, are very much important. Similar like what we found in Staphylococcus aureus is called small colony variants, and Aciniobacter are also able to produce a lot of small colony variants. These are the small colonies which are potentially resistant. They always have different types of variant cell types within a cell, which can produce highly resistant behavior. And of course, we keep on analyzing whether it can able to produce biofilm. This is a tube which shows the ring is called biofilms, and these biofilms some bacteria make more, some bacteria make less. So we have to see how much biofilms are being produced every bacteria of Acinetobacter baumannii. And further, we do the confocal microscopy to understand what is the volume of these biofilms, are whether it is not the volume and thickness of the biofilm. We keep on measure and try to see what are different genes which are important in making these bacteria to become more biofilm other stuffs. So continuously we also try to understand the reflex mechanism, which is the primary mechanism through which bacteria resist. So we see whether these bacteria are able to make a reflex. So we quickly do sort of a growth reflex or expression. The expression is basically just to understand the real-time expression for certain genes which are the known predominantly found in Acinetobacter baumannii. Whether these genes are there, definitely we can see that certain type of resistance is going to emerge. So these are the basic elemental exercises one has to understand always before going into the detailed study, because just see whether all the bacteria, whatever the bacteria, whether it is able to have antibiotic resistant phenomena first, and second is able to have a reflex mechanism primarily in operative, and we have to see the trademark genes which are being expressed or not. So these, importantly, one has to keep on analyzing for a set of isolates. And also we do routinely the phenotype microarray, which is nothing but to understand the metabolic profiling of a bacteria, which can utilize a carbon, nitrogen, and different sources. So based on these analyses, what we do is we try to identify a some hypothetical protein which are being expressed in a set of clinical isolates, and those genes are going to be potentially important in studying further, like in detail. That's what we do, and simultaneously whatever we have sequenced more than 20-30 genomes of Acinetobacter baumannii to understand how these resistant genes are now distributed, or how whether we can be able to get some idea of a contribution of any silent gene which can now make these bacteria more resistant. So interestingly what we found is the two genomes which we sequenced out of the pool of more than 20-25 genomes, we find that two interesting sequence types, that is novel sequence types, which is reported for the first time in India. And these two interesting sequence types, both are resistant. One is highly resistant. So you can see the ST195 is highly resistant, and ST128 is a sequence type, and ST128 is a moderate resistance. Though these both are also resistant, but it has a genetic properties completely different in one and other. So because if you see the highly resistant, the protein analysis, specifically the core in which I said before, we have a specific mutations. So once you have a mutations, you can see the structure is completely changed over here when compared to the one which is a sensitive where there is no mutation. So these are some of the elemental differences we can always see in a battery of resistant genes. So in order to understand the precise mechanisms, so we know largely reflex pumps, outer membrane proteins, like modifying beta-lactam enzymes. So these are the elementally, if you have it in any bacteria, the bacteria become resistant to different classes of antibiotics. So it's all known. So it's what our aim was just to find any new protein or any new, we need to identify a target. Basically, the drug, whatever the drug we are going to discover, it should not be have a resistance problem later. So on understanding, because on analyzing the whole expression profile of a bacteria, we could see a lot of genes which are not characterized in Acinetobacter baumannii. Acinetobacter baumannii is having 3,000 to 4,000 genes, and all the genes are not responsible for making the bacteria more antibiotic resistant. Definitely a lot of genes are involved to make a bacteria more highly resistant. So in that case, we identified one gene which is called outer membrane reflex protein, which we termed as ABUO. So whenever we identify any specific gene, what we do is we try to characterize the whole gene in terms of phenotypic as well as a genotypic. So the phenotypically, what we do is we try to express heterologously and see how whether it is these genes are contributing for antibiotic resistance. And secondly is we make a genetic knockouts. That means in Acinetobacter baumannii genome itself, we delete this gene and try to see whether it has any role on antibiotic resistance by doing a simple kidney bearer assay and lot of other biochemical assays to find whether it has a role on antibiotic resistance. So since in that way we identified that this gene is having a major role on antibiotic resistance as well as osmotic stress response. So it can, if you delete this gene, you can always see amikacin, carbacin, ceftriaxone, meropenem, streptomycin. These are the antibiotics which are basically it can give resistance towards these antibiotics by this gene. So and of course that all done in a single bacteria, right? And similarly we also try to take a different clinical isolates of Acinetobacter baumannii, because all these Acinetobacter baumannii is having a different resistant type of patterns. Like not all are highly resistant, some are low in the medium resistance, some are very high resistance. So different types of a resistant gene Acinetobacter bacteria we take, and try to make every individual mutations in all the different genomes, and try to ask the same question whether the gene, if you knock out, whether we could see any difference in antibiotic resistance. So directly we can show this role of this gene in antibiotic resistance by making knockouts. So that overall what we found is this gene is having a major role in antibiotic resistance, especially for the beta-lactams as well as aminoglycoside. It can have a role. So of course, the reflex pumps are the one which are found abundantly in many bacteria, especially in Acinetobacter baumannii. You can see the spectrum of reflex pumps, the largely different families, which I said like more than all the 5 families. You can see in the number, almost 40-50 genes are there, and out of all the 40 genes, like certain genes from our lab we have identified its role, especially on antibiotic resistance. The genes, whatever, different families of reflex pumps like MFS, SMR, these are the different types of families of reflex pumps, and each has its different characteristics. Some are small, some are big in terms of 2-3 protein mix of reflex pumps, and these reflex pumps are the robust one which can throw a lot of antibiotics. Lot of antibiotics means different classes of antibiotics. Not only antibiotics, it can throw amino acids, biocides, and other nutrients, because these reflex pumps are in bacteria is meant not only for throwing antibiotics. For normal survival of a bacteria, these reflex pumps are necessary. So, a lot of ion movement is there, nutrient exchange will be there, and a lot of pathogenic factors are, keep on moving from inside and outside. So, largely, these pumps are called as a broad substrate specificity. That means it can throw not only antibiotics, biocides, or dyes, or disinfectants, or detergents, many things it can throw. So, and we know these pumps are always, if it is expressed too much, then it is a problem for any bacteria. How these pumps are being expressed is an important question, and during our study we identified one a pump which is always expressed in different clinical strains of Acinetobacter baumannii, which belongs to R&D type of a membrane protein, because all these reflex pumps are membrane bound protein which always have certain roles on antibiotic resistance, and primarily we do in our lab is just you take any gene, we try to ask whether this gene has any role on antibiotic resistance on Acinetobacter baumannii by making genetic knockouts. We make a lot of genetic deletions on a chromosome of Acinetobacter baumannii and try to ask the same questions whether it has a role on antibiotic or antimicrobial resistance in terms, so something like that. So where we can see it contributes to the antibiotic resistance, and similar way other reflex pumps, because these pumps are different types of families also. So different families, we need to characterize all the pumps, otherwise we don't understand what are the pumps are involved in contributing antibiotic resistance to Acinetobacter baumannii per se, you can say. So we keep on try to challenge each and every gene and try to ask a similar question whether it has a role or not. So generally all these we take these genes, we try to make a knockouts on a genetic map where we can do a lot of functional and biological assays to characterize the involvement of these genes on antibiotic resistance. So almost all the genes, whatever we have and for reflex pumps, we identified its role on antibiotic resistance and also some of the physiologically relevant stress conditions like osmotic or oxidative stress. These are the stress which always you can encounter by a bacteria in a human host or in the environment also. So because the stress bacteria always exposes to different stress conditions, during that stress a lot of other genes are expressed which basically manifest in antibiotic resistance always. So what we know so far is the presence of resistant genes in the Dr.N.G.P genome. We try to explore the functional role of these genes, but these all sporadically we can see here and there, but all these are coordinated by some sort of regulations. So the bacteria has all apparatus. It will independently operate under certain conditions only. It will these genes can be switched on or switched off. So these switches are modulated by certain type of a signaling proteins called as a two-component systems. So two-component system is nothing but two proteins are involved in communicating, manipulating the whole regulations. So in bacteria, specifically in some of the Dr.N.G.P, you can say 20 different types of proteins are there. That means 22 component systems are there. Always, they can each protein has specific signals, not suppose if bacteria undergoes a very low pH, certain two component systems are expressed, not all 20 or 30 is being expressed. So these one or two component systems play a major role in regulating whole cascade of genes just to keep the bacteria happily stays in different conditions, be it low acidic conditions or maybe at alkaline conditions or maybe very high antibiotic conditions, which leads to oxidative or some sort of a stress conditions. So under the stress conditions it can happily stay nicely because bacteria is equipped with a lot of background proteins or signaling proteins which can have a major role in modulating the antibiotic resistance behavior like that. So what we do is similar like antibiotic resistant determinants. We try to also characterize in detail, understand whatever the gene, because how do you pick these genes, whether these genes are important for us, the Dr.N.G.P physiology, that we do normally try to understand by host pathogen interactions and try to see what are the genes are being expressed when we try to expose to the host, and those genes we have to target and which will be try to completely characterize in terms of biochemical, biologically we have to characterize. So that means we have to first identify, because all these genes which are uncharacterized previously, we have to first identify its completely the role of this gene with respect to antibiotic resistance in some of the Dr.N.G.P. Then only one can think whether it has a direct or indirect role. So we have characterized many genes like that. These are some examples which I am showing it. This is now quickly we have to do, because these are the protein signaling proteins are nothing but phosphorylated proteins. So we have to do some sort of in vitro autophosphorylation kinase assay, that means phosphorylating whether it can undergo a phosphorylation or not. So those things will tell you this protein is functionally active or not. So once you do, you try to make knockouts, genetic knockouts in a chromosome of the Dr.N.G.P, and try to ask the same questions whether knocking out of a specific gene it has an impact on antibiotic resistance or virulence, something like that. So we do routinely like these type of assays and creating a lot of knockouts on chromosomes and try to challenge Acinetobacter and try to understand the whole process. So in that way, we identify a lot of unknown protein that is called as a two-component protein. We are known to have a role on antibiotic resistance. Some are very specific towards calcium. That means within a calcium, if you have it in a medium, these two-component systems are being expressed better than any other two-component systems, and also not only expression, that also makes antibiotic resistance. So some are two-component systems are very specific to regulating beta-lactam type of antibiotic resistant genes. So like that. So we can easily understand certain genes function by manipulating individual genes or understanding in detail, then only we can score the functional relevance of this gene Acinetobacter baumannii. So in addition to that, bacteria is also having a different set of specialized proteins called protein kinases. That is why these protein kinases are important, because these are the straightforward drug target. That means what it means is whether be it bacterial tyrosine kinase. Now I can say, for example, here we choose in detail study further because it doesn't have any eukaryotic homolog. That means human have a lot of tyrosine kinases, the huge family is there, but whatever bacteria, specifically in Acinetobacter baumannii, we have a tyrosine kinase which is discovered recently that bacteria also have a similar type of tyrosine kinases, which is very different from human. That means once you have a target, this is a straightforward target for making any drugs. So once you understand these kinase protein in detail, probably one can develop any inhibitor against these kinases. We can develop a new drug specifically against Acinetobacter baumannii. The simple reason is, first, it doesn't have specific homolog on humans. That means straightforwardly we can use for treating Acinetobacter baumannii in human populations. So what are different, what are tyrosine kinases? Because the interesting fact of tyrosine kinases are in gram negatives, you can always see as a single polypeptide chain. In gram negatives, positives, you can see two proteins are there, having similar function. That means in gram negative you can see a single protein which encodes for the tyrosine kinase, but in gram positives you need to have a two protein to have a functional protein. So what we see is the signature mark of any protein, that is tyrosine kinase. Protein is a tyrosine cluster at a C terminus of a carboxy terminus of a protein. So if you see big 10 or 15 tyrosine clusters are there, and these are the signature marker for any tyrosine kinase protein. You can see. So what we do is try to, because we identified this by serendipity, these kinase we identified during our study, and just because this protein is not at all characterized. So what we thought is initially whether we identified based on structure and sequence basis. We thought maybe whether this is now having functionally active or not. So we cloned, expressed. So everything we do, try to understand first whether it can have functional by using radioisotope ATP source. So based on that, we can see now addition of phosphate is happening to the proteins, so it becomes autophosphorylation. So this is functionally active, and of course we have to make a lot of genetic knockouts importantly, because seeing only gene on Acinetobacter baumannii doesn't guarantee for antibiotic resistance problem or resistance interactions. So we have to really characterize in detail. So we make a lot of knockouts and we do a series of assays, biochemical and biological assays. The important point what we see after deleting this gene is normally Acinetobacter baumannii is a non-motile, so it doesn't make any flagella, but it has a small pilus-like structures because these you can see in this electro micrograph interaction between two bacteria happens through the pilus always. So a lot of these are the wild types. That means normal Acinetobacter baumannii, the gene is not deleted. You can see a lot of interactions are there. The moment that you delete the tyrosine kinase, the pilus is completely gone. That means the shape also become little bit pleomorphic, and the moment you make a double knockouts, that means 2, 3 other phosphatase gene is there adjacent to that. That also if you knock, you can see like a complete removal of a pilus. So that means it completely the loss, the gene which is having a role in interactions are completely also there. So that we are related through scanning electron microscope or transmission electron microscope. So these are different. You can see here these are wild type very globally. A lot of background we can see the pilus-like structures. The moment knocked out, the cells becoming bulging, and the complete background is the pilus structure is lost. That protein which is formed is also lost. So that way you can see not only loss, interestingly we could see the antibiotic resistance towards a certain group of antibiotics. So cephalosporins. In the wild type you can say the MIC is now 256, and if it is knocked down the genes in different combinations, the MIC also goes down. So it's not something like the gene is not only having antibiotic resistance role, also it has a significant role in modulating the pilus formation, which are very essentially required for transfer of genetic material from from different bacteria. So that's interesting what we got, and once these genes are involved in certain things, we try to understand in detail what are the downstream genes. At the global level, one has to understand the complete background of once you knock out, what are the different out of 4,000 genes are becoming responsive, or how these genes are being modulating. So by using different techniques specifically transcriptomics, where you can see a lot of changes at RNA level, or at a protein level you can use proteomics. We have done proteomics and mass spec quantitative proteomics, where we can have a set of genes which are responsive directly against tyrosine kinase. So we make, based on that assumptions, we make a central metabolic maps. In the left panel you can see normally in Acinetobacter baumannii, these are different metabolic genes and metabolic pathways where different genes are, all the 4,000 genes are encoding here, and these colors are nothing but the genes. Once you make a knockout, you can see the changes of different colors. That is basically some genes are upregulated, some genes are downregulated. The overall metabolism is completely hampered, and if you see in the down panel, the energy metabolism is completely gone. That fermentation is the gene is down. That means down regulation of, you can see down regulation of efflux pumps or the transport is being observed if you knock out these. So the degradation, we can see a lot of amino acid degradation is going down, carbohydrate degradation is going down. So basically it has a significant impact on Acinetobacter baumannii physiology per se itself. The moment you make knockouts of tyrosine kinase, you can see a lot of functionality is completely drastically reduced in terms of the overall bacterial metabolism. So we are currently, trying to understand since these are promising drug targets. So we are exploring certain inhibitor against designing against these specific tyrosine kinase. So that's a part which we go in it. So point is one can easily try to understand in detail what are different genes are involved impacting the antibiotic resistance, because antibiotic resistance is not a single person job. Basically, the whole bacteria has more than 3,000 to 4,000 genes. All these major set of genes are involving in impacting similar type of a phenotypic expression of antibiotic resistance. So the second part I'm coming is essentially very much important at the current context, the One Health. What is One Health? Why it is very much important? So One Health, because it's coming back, is almost all the emerging diseases, whatever we have seen in humans, are zoonotic only. That means all coming from animals. So more than 60%, you can say 80% are potentially problematic, and the recent emergence of a coronavirus is from animals. So many are there. This is a very much documented. So, and the One Health is, the concept is nothing new concept. So back in history, if you go back, the Segregation and Quarantine is the best book by Columbia, where it is advised the people and animals should be kept separate on farms. So this is all background, Hippocrates said that the concept of public health depend on clean environment. The environment should be clean. So the whole catchy of One Health started when Roloff started putting a book on the clear understanding between animal and human medicine. There is no division. So both are animal and human medicines are almost same. So the whole One Health started when Sir Kelvin put up the concept of one medicine and one health, which clearly indicates that idea that the health of a people is connected to the health of animals and our shared environment. So this is a worldwide strategy where one has to, all the disciplines will be collaborations and communications in terms of a human, animal and environmental safety. So like a history back, if you see, bacteria is evolved 4 billion years ago, but humans are just 200 years back. And humans are made up of what? All bacteria only. Almost 90% of bacteria, the whole human, we have it, be it in all the organ is populated by bacteria. And you can say weighs almost like 2 kilograms of bacteria we are holding it always. So, so the thing is, these bacteria are evolved 4 billion years back, and these are present now everywhere. That means throughout the environment, the same bacteria which you can see it on human, you can see it on animals, see it on environment, see it on plants, see it on water, everywhere. So the whole microbiome is completely shared among the whole animals, plants and environment as well as humans. So we have these the bacteria is already shared, and the genes are sits in a certain bacteria. Definitely you can now see the transmission of genes always happening rampantly in the environment. So there is these are the movements of antibiotic resistance. If you observe in the clinics, definitely you can see it in the environment, and if it is environment, it keeps on moving from environment to clinics or animals, plants, everywhere. So you can see antibiotic resistance keep on migrating from different hosts, and the moment you start using more antibiotics in the presence of selective pressure, that is antibiotics are more on environment, the development of antibiotic resistance will be more. So the current paradigm, if you see for human diseases, always we do disease surveillance, then investigate what are the different agents, and based on the different antibiotic resistant bacteria, you start doing the treatment. This is the general paradigm which we follow at clinical settings. The once the moment you put One Health perspective, you have to basically screen the environment, like in terms of animals or human or environment, the whole thing. We have to do a surveillance study and try to predict whether you are going to get any specific outbreaks or any specific disease. So based on that, we can make prevention strategies. So if you come across, I will go through the important certain parameters where we can see the consumption of antibiotics in a food animal is drastically heavy in the terms unimaginably heavy. You can see in China the big circle indicates more than 50,000 tons of antibiotics are being used. So it is projected by 2030, 200,000 tons of antibiotics are being used throughout the globe. So it's like the antibiotics are being misused more efficiently in the production of animal. Excuse me. So in that case, India, we have more of antibiotic resistance. Antibiotics are being used at animal productions. So be it chicken, pigs or cattle, there is a clear-cut threefold increase of resistance always observed in these animals. And this is the paper which they identified in 2018. But definitely we have the 3 to 5-fold increase of antibiotic resistance always in the food animals. And the demand for poultry increased from 2 to 7-fold because this demand is always going to be more in India, specifically because consumption of poultry is more in lower income countries, especially China and India. The interesting part is China is using more antibiotics in the animals when compared to the humans. So the bulk of antibiotics are being used for as a growth promoting factors in animal production is really very severe. So because of that, you can always we have seen the colistin resistance. Sorry for the spelling here. Colistin is the one antibiotic which is a last resort of antibiotics used for gram-negative infections. The bacteria become resistant to carbapenems. There is only left out option is either polymyxin or colistin, and this, since it is nephrotoxic, people they don't administer it regularly, so it is a last resort. And whatever we know so far is chromosomally now these resistant determinants are sitting. The colistin become resistant because of certain chromosomal bond genes, but because of lot of use of colistins on an environment, especially in the pig production in China, you can see the emergence of plasmid mediated colistin resistance, that is called mobilizable colistin resistant MCR1. Similar like MDM1, they have identified MCR1 some year back, and definitely now almost a lot of variants of MCR colistin variants are being observed on different bacteria, and throughout the world you can see colistin resistance MCR genes are there on different bacterial hosts. And the Punjab, currently this is the biggest problem in Punjab, where poultry farms are being profusely used, lot of colistins and lot of antibiotics, tetracycline, other stuff. So this is the report which says whatever you isolate from poultry farms, be it E. coli or Klebsiella, it is 100% resistance towards the new clinical line of antibiotics. We can see 100% of resistant. So they basically, whatever the isolate you got from these poultry fecal material, we can see resistance. So based on that, we proposed one grant from our team where we got Bairak and Bill Gates project, where we try to understand the metagenomic approach of One Health surveillance of antibiotic resistance in poultry setting, especially. This is Asia's biggest poultry setting, which we have it in Haryana, where we try to understand the role of poultry and farm workers and family members of the farm workers. So we want to understand what are the diversity or whether these AMR resistant determinants are able to migrate from animal to human or human to animal. So based on our initial studies, because of some metagenomic approaches, we have extensively studied on that. We understood the sharing of microbes are there between, excuse me, between poultry and farm workers. You can see the difference, and importantly what we found in the poultry setting is a lot of pathogens which are found in the clinical settings are found on the poultry settings. Excuse me. So coming back, a lot of success stories are there in One Health, and few I can mention over here. One is in US and Mexico, the brown dog ticks, which carries a germs called, basically which causes Rocky Mountain spotted fever in people and dogs. So the dogs in Arizona started roaming freely on the streets, and people are getting infected. A lot of people are dying because of Rocky Mountain spotted fever, and initially they are very clueless, and later identified it is coming from dogs, and dogs were completely quarantined in terms of application, using the pesticide applications around homes and other stuff, and try to contain the germs on that dog, and thereby after say in terms of 4 months or time, 90% of dogs were tick-free in the community, and thereby they reduced the Rocky Mountain fever, completely went down from the community. So this is the way one try to understand the One Health impacts on disease control, and thereby one can do a lot of examples are there. The classically we can say Nipah virus, they have done it in Malaysia. Definitely, in India also, in Kerala, they have done a similar approach where using the One Health principles, they contained the Nipah virus emergence, because these are infections doesn't have any vaccine, so it's a very lethal. So coming back is we know these antibiotic resistance always have a problem, but we have to follow certain quick solutions which we have to follow ourselves, like in terms we don't do proper hand washing. Hand washing is an important custom where our old school of thought in our home itself, our old parents used to say wash your hands then come for eating. So the practice is completely which we don't follow it right now. So because of that, a lot of diarrheal infections, many reports are there. And specifically WHO, they have a specific hand washing day in October. So we should not always wait for hand washing day, but we have to follow the principles of hand wash. It's not like something you keep on quickly wash your hands. There is a procedure which recommended by WHO. We have 6 movements, in terms of washing the using a soap, how to wash, and in different places, because certain places of hands are highly occupied with a lot of bacteria because we don't regularly wash. So I think if I just 1 second, I will play a video on hand wash, how to do. hand wash is recommended by WHO. One second. Can you see my video now? One second. So I'm just closing down here. So overall AMR requires One Health Global Solutions. So thank you very much, and thank you very much for patient listening. I will take up my questions, and I once again thank the organizers for having such a wonderful interactions. Definitely next time I will come there. My apologies for not joining there, and thank you very much.
Thank you, sir. It was indeed a very elaborate and a very interesting section. I'm sure when the students hear it from the voice of a scientist, it's a totally different world altogether than the way the academician projects it. So I'm sure my students would have enjoyed, and maybe they have a couple of questions. If it's possible, maybe we can take some time out to answer them.
No problem. Just anything if you want to ask, you can ask me, no problem.
If you have any questions, you can ask, sir.
Right from where you are.
Presentation, sir, it was very interesting. I have one question.
Yeah, please.
Sir, for AMR, what may be the possible solution? New drug generation or alternative therapies? Like,
Okay, this could be very interesting. Nobody has no specific answer for this. So new drug is always important, because whatever the alternative therapies if you put, bacteria is going to become resistant for anything. So we have to devise new drug. Bacteria should not become resistant towards that also. That is the whole game which people are trying to see. So whatever the drug you make, bacteria should not become resistant, because the whole pharma industry is not coming forward in terms of developing new antibiotics. The reason is they make new antibiotics. Antibiotics takes a lot of long process, 15-20 years it will take the new drugs. Once you make a drug, the bacteria within minutes of a time develop antibiotic resistance, because we don't use it very efficiently in terms of antibiotics. Whatever the drugs we use, we have to use it very optimally, not to misuse it. If you start misusing, you will develop resistance. So if you say we have to use always identify new drugs, that is the one way. And some parts where we can, wherever possible, we can use alternative therapy also we can use. So it's not straightforwardly we have to use always new drugs or develop new drugs. So whatever effective way we have to do in that way. So it has to go hand by hand always. Both we have to do, new drugs development has to be keep on going, and develop some sort of a new alternative therapies. People keep on coming up with fast therapies, combination of different antibiotics, combination of certain essential oils, many factors are there. So people are putting vaccines also. So it's we have to explore more. Okay.
So I have a question from my side.
Yes.
Whenever we talk about antibiotics, we use the type of antibiotics. Antibiotics we talk about the different locations in the state of AMCID, different parts of we talk about the different type of antibiotics. Do we have any specific antibiotics that work against flippage?
Work against?
Flippage.
Yes. I think this is one target which we initially studied during my postdoc itself. We were in this customer which is called MSBA gene, which now flippers are the one. We don't have antibiotics against the target, but people are using it because flippers is the one which is it is difficult. It's all sort of a membrane bound protein, and one has to develop a lot of, because the structure of a protein is not well studied. Now we have a structure on that, and mechanism is previously not known. Now we know how these ions or antibiotics are able to flip between inner or outer membrane of a bacteria. So targets, people are targeted that flippers, but it is all in the initial stages. But I don't know, people will use that because drug cannot enter the cell, right? So drug has to go inside first. The barrier is first is outside. The drug you make, it has to go inside the cell first. Second is it has to reach into the intracellular components, specifically flippers. So, for example, if you start developing antibacterial agents against flippers, we need to have certain lot of strategies. But few people also started working on that. They have a lot of variants on antibacterial variants against flippers. It's all under production only. It's all under production. Very difficult to say which one is going to have great practicality, implication in terms of usability. Later stages only it will come, but at least people are not leaving any gene, whatever the gene which plays a major role on bacterial growth or physiology or antibiotic resistance. People are targeting it, definitely.
Thank you.
Okay.
Good morning, sir. Thank you for the presentation. Thank you, sir. Thank you for the informative session. And my question is, if it is possible for the multi-drug resistant organism like escape to have a common driving factor that separates from other bacteria, the other bacteria that lacks to take resistance, is there a mechanism that separates these pathogens?
Can you say it again, because your voice in between breaks? Can you repeat the question, please?
Sir, if it is possible for the multi-drug resistant organism like escape to have a common driving factor to get resistant to these antibiotics that separates these pathogens from other bacteria.
Your question is still not able to get your voice. You said escape and between other bacteria. What is missing?
But do we have a methodology to identify the multi-drug resistant strains from the other pathogens?
Oh, okay. So in a pool of yes, we have a lot of methodology to identify. Normally, human gut, you have commensal, right? Commensal is sort of less resistant, or we can say sensitive towards all antibiotics. So, the moment you start isolating antibiotic resistance, how do you isolate is basically you try to either use phenotypic method. One is you start culturing on a certain plates where we can have drug on the plate, or maybe the substrate which will show that this bacteria is going to produce antibiotic resistance, or maybe we can use genetic markers where all the resistant markers, if this bacteria is having, then we can term this AMR. So the basic purpose of initially how we can do is like we can do quickly antibiotic Kirby-Beyer assay, that is disc diffusion assay by using a different antibiotic disc. Based on that, we can see whether this bacteria is antibiotic resistant or sensitive. So based on that, we can easily differentiate. Good morning, everyone.
Very interesting. And I would like to take the question next. What causes this mutation? Is it whether prolonged exposure to this same medicine which is used by her, or else some sort of spontaneous mutation which is occurred intensively due to some gene which are present in themselves?
Okay. So this is very interesting. You asked it. At least evolutionary wise, bacteria, one has to have corrective pressure in the terms of any antibiotics. Any antibiotic, if you start using in your culture room, in your lab itself, you start growing E. coli, and you directly add ampicillin daily, maybe you add less, keep on passaging the cells for every 1 week. After 1 week, you can see the day 0 is ampicillin is 0 MAC, the day 5 MAC will be more than 256 microgram. That means bacteria is able to make a lot of mutation. That means spontaneous mutation arise due to selective pressure of antibiotics. So be it any antibiotic, not only specifically carbapenems or beta-lactams or maybe fluoroquinolones. Most of the bacteria are much susceptible to a lot of antibiotics. So that is where we can see antibiotic resistance during treatment of in the clinic setup. Patients are undergoing a lot of antibiotics. Antibiotics, what we are eating for suppose for any fever, you say doctor used to recommend almost 2 mg or 1 mg per day, per time. That means 3 times in a day you take almost 3 milligrams of ampicillin or amoxicillin along with clavinic acid you take. Say you are pumping almost 3 to 4 grams of antibiotics. This is at community settings, community level. But hospital level, people used to add a lot of antibiotics in for the treatment purpose. It's not like something which we do milligrams. We do it in grams by body weight. So you infuse a lot of antibiotics. That means your body is having a lot of antibiotics. The selection pressure is always heavy to mutate. The normal bacteria become resistant because they have to sustain the antibiotic pressure, right? Otherwise, the bacteria will die. Instead of dying, it has mechanism through which it can become resistant. So it will make a certain portion of a genome become mutation. That means some genes we know, some genes are more liable to mutations. Once you have mutations, then the bacteria become more resistant. So your question is you add any antibiotics, definitely bacteria become resistant after a long time. It is called spontaneous mutation, you said it rightly. So that's where you that mutation is not random event. It is specifically on a certain bacterial genome, and it will make that bacteria to become more resistant.
Thank you, sir.
Hello, sir. Your session was indeed very interesting. Thank you for your time, sir.
Okay.
So my question is, you mentioned that phage therapy is one of the alternative solutions for antimicrobial resistance. Is it a promising solution yet? And if not, what are the gaps in its research?
Yeah, the phage therapy is not something new. It's very old because in Europe and Western countries, people use phage therapy, and even in US, like we don't have any antibiotics, specifically in terms of Acinetobacter baumannii, because it is a globally number one pathogen where you can see no antibiotics are there. So we have to develop antibiotics against this Acinetobacter baumannii, because the moment Acinetobacter infection becomes multi-drug resistance, nowhere in this world we have treatment options. So that's where people start using phage therapy. Success stories are there. Then the limitation of phage therapy always is phage therapy is not a very broad spectrum. That means it has to be very specific. So we cannot use routinely like what we do for antibiotics. Start taking antibiotics, and you can become cured. So like in phage therapy, we can use more than once or twice. It becomes more, and it won't more always. It won't always well. And specifically, it is a strain specific mechanism. Phage is always try to like specific against skin and also we cannot use routinely like what we use like antibiotics. That is a major drawback. But people are what they are coming up is phage is able to make some sort of lysine. They are trying to use those protein or the lysine protein, which is in combination with antibiotics. People are trying it in that way. So it's not something always advisable that phage therapy is going to come up in. It's not going to replace the antibiotics. Impossible right now, and it may be full in terms of patient specific or case specific. It's not widely we cannot use as phage therapy.
Thank you, sir.
Thank you.
Any questions?
Any more question that you would like to ask, sir? There are no more questions, then we can wind up the session, sir. I don't need to tell you how interesting your session was, because it's very difficult to keep an audience at pin-drop silence, which we have been observing for the past almost 1.5 to 2 hours, which means that your presentation was extremely useful and interesting for the students. Starting all the way from transmission to mechanism and talking about the most interesting up-to-date technology, which is gene knockout technology. You have tried to cover most of the concepts in AMR within a given short span of time. Thank you very much, sir. Thank you so much for joining us online. Hope we will have you with us outside very soon.
Okay, thank you. I think thank you. Yeah, some other time I will visit your college, but whatever you can go to the Intech website, or we do a lot of programs here in terms of short-term, long-term. So students can always apply and try to catch up the whatever new things are happening at scientific frontiers. So we do routine animal experimental procedures, a lot of workshops, hands-on. So whatever is coming, I can share with you with your department head and principal. So you can always apply and try to enjoy the rich experience whatever we can offer you. Okay, thank you very much. Thank you very much for all the things.
Now we have a formal vote of thanks, sir, which will be delivered by Ms. Vidisha from 2nd BSc Biotechnology. Over to Vidisha. Good morning, everyone. It's a privilege to stand for you today to deliver the vote of thanks at this insightful session on antimicrobial resistance and One Health perspective. I extend my heartfelt gratitude to our esteemed speaker, Dr. G. G. Rajmohan, who has shared his valuable insights on this critical global challenge. Your expertise has deepened our understanding of antimicrobial resistance and interconnectedness of human, animal, and environmental health and then One Health approach. Thank you, sir, for the valuable session.
Thank you. Okay, thank you. I'm leaving now, okay? Thank you.
Okay.
Okay, thank you very much. Okay.
Now it's time to refresh and recharge. Refreshments will be provided in underground. Please be here back within 10 minutes. I repeat, refreshments will be provided in underground. After having your refreshments, please be here within 10 minutes.
Manish.
Hello, students, maintain silence.
Check.
SP sir door கிட்ட நிக்காதீங்க.
Hello.
ஒண்ணுமே கேட்கல.
Check.
போனா இங்க பாரு. SP sir.
Hello. Hello. Check.
போனேன். அப்படியே எடுக்க முடியாது.
Check.
Check.
எங்க போறோம்?
ஹலோ செக்
நான் கால் பண்ணி வர சொல்றேன். உட்கார்ந்து
ஹலோ செக்
Students are requested to assemble inside the hall. Let's move on to the technical session 5. It explores the topic of monocyte heterogeneity in pulmonary tuberculosis. We request the keynote speaker and session chair to occupy the dais.
now invite Ms. Aruke Sterling from First MSc Microbiology to give an introduction about the session chair, Dr. P. Arun from the Department of Biotechnology.
I stand before you today on behalf of Department of Microbiology to introduce a person of great wisdom, our Honorable Chair, Dr. P. Arun, Professor, Department of Biotechnology, who has 15 plus years of teaching experience and 13 years of research experience. Dr. P. Arun had completed his graduation in BSc, MSc in the field of biotechnology at our own institution, Dr.N.G.P Arts and Science College. M.Phil at Periyar University. He also completed his MBA in the management at Bharatiyar University and Ph.D. in biotechnology. He had his dynamic participation in publishing national, international journals, conference presentation, workshops and faculty development programs. He is proactively being the member in the board of biotechnology at Dr.N.G.P Arts and Science College since 2015 and at Hindustan College of Arts and Science, Coimbatore since 2021. He was awarded with fellowship from DBT, India for attending training program in CFTRI, Mysore. Dr. P. Arun had his incredible contribution in gene bank submission of the species Acinetobacter johnsonii strain along with his colleague Dr. R. Surya. It is said that great leaders recognize greatness in others. With this in mind, I now invite our wonderful chair, Dr. P. Arun, to introduce our special guest, Dr. B. Ramalingam. Sir, please.
Good morning, everyone, and thank you for the introduction. I extend my heartfelt thanks to the organizing team for granting me this opportunity, and very happy Science Day wishes to everyone. It's my honor and privilege to introduce our esteemed chief guest, Dr. Ramalingam Bethunayakan, a distinguished scientist and leader in the field of molecular immunology and infectious disease research. Dr. Ramalingam is currently serving as Scientist F and Senior Deputy Director at Department of Immunology, ICMR, NIRT, Chennai. With an illustrious academic background, he holds Ph.D. in Biomedical Science with the specialization of Immunology from the Tuberculosis Research Center, Chennai. His journey of scientific excellence spans over 26 years, focusing on innate immunity, infectious diseases, and tuberculosis research. His remarkable career includes international exposure as a postdoctoral research scientist at Columbia University, New York, USA, and as a researcher at the Institute of Pasteur Lee, France. His pioneering work is understanding host immune responses, vitamin D as an immune modulator in tuberculosis, and molecular characterization of monocyte subset has significantly contributed to the advancement of infectious disease research. Dr. Ramalingam has received the Palaniswami Re-Entry Fellowship from DBT, Government of India, and various research grants from NIH USA, DBT, and ICMR, totaling approximately 19 crores, which have been completed, and as well as ongoing projects around 13 crores. He has authored 89 high-impact publications and 5 book chapters, and Sir is playing a vital role in mentoring young researchers. His dedication of translation research and public health improvement makes him a true inspiration for aspiring scientists. We are truly privileged to have him with us today, and we eagerly look forward to his valuable science. Let's extend a warm welcome to Dr. Ramalingam Bethunayakan. Sir, we welcome, sir.
We now request Dr. P. Arun to We now request Dr. P. Arun to honor our keynote speaker with a shawl and memento as a token of gratitude. Thank you, sir. Sir, the dais is yours.
Next point. Is it? Next to the previous one. The lights not off. Yeah, fine. So, good morning, everyone. So, I'd like to thank the management and the organizers for giving me this opportunity to meet you all and to share some of our research with you. So I hope it is all microbiology. So microbiologists are well versed in about these basic pathogens, and here today we are going to discuss about some of the very well evolved pathogen, which is Mycobacterium tuberculosis. And we are much more specific into the subset. So everyone knows that mycobacteria resides into the monocytes or macrophages. So I think you should all know how these two evolve together. So with that, so there are a lot of questions arises how each one is competing between for their own survival, for which we'll now go into the details. So TB is not a unknown disease. It is a common disease which is being known for centuries, and it can affect the pulmonary system, not only the pulmonary system but also the other parts of the body and lungs. So they are all pulmonary and extrapulmonary. And India ranks the biggest among the global field, where it is very huge. The system is, and it ranks around the top 8 countries across the world. And these are all the basic symptoms. You should be aware of it. Fever, fatigue, weight loss, persistent cough, blood in the cough, and night sweat. So whenever you have anything which is persistent for more than a week, you should think about this TB disease. So this disease is presented in a various spectrum, where you can see it as a latent TB or active TB, or even in a worse called as MDR TB. So this spectrum accompanies everything. And now if I ask you one question, how many of you in this audience are infected with Mycobacterium tuberculosis? So can you say what will be the proportion? Any idea? So we think we are all very clean. We do not have any pathogen, and we are not having this TB. But the truth is, more than 40% of this population is infected with TB. We do not have the symptoms. We will be having the symptoms as we grow old or when immune system drops. Malnutrition, comorbidities like diabetes and HIV, and as you progress in age, senescence of immunology, these things will drive your pathogenesis. So in a normal circumstances, otherwise 40% of this population is infected with MTB. So that comes into the initial stage of the spectrum, where this is called the latent TB infection. Then comes the drug sensitive, which is an active form of TB. Then comes the drug resistant TB. So this distinction between these categories is mainly in terms of host immune response and also the host damage, which is mainly rely on this immune response, inflammatory reaction, and the mycobacterial burden. So these three conditions are very important to drive the entire spectrum of the disease. And coming to the pathogenesis, host-pathogen interaction is very, very important, and you should know which decides the fate of the pathogen. And so for the successful host immune response, our body elicits most of the immune parameters, particularly the pro-inflammatory cytokines, and also the adipokines and other activation of monocytes. Until unless the activation of monocytes happen, this cannot be fought under the host immune system. And for the successful pathogen response, it awaits with the hyperinflammatory response and the cytokine imbalance, which is where the pathogen is successful. So the inflammatory condition is very trivial in order to switch your condition from a successful pathogen to a successful host. So now everybody is fighting for this TB because we want to eliminate this, but there are some drawbacks where our researchers feel there are some limitations and what the government and policy is working towards it. So the main thing here is a pool of undiagnosed TB population. As I said, 40% of this population is latent TB, but nobody is having an awareness of that. We do not have a proper tool to measure it. So that is why this 40% of this population is not being properly diagnosed. An emergence of multi-drug resistance. As the microbe is evolving rapidly, they come up with new drug resistance. So the antibiotics fail to contain them and also to progress into the research. And limited assays, diagnostic assays, and then lack of assays to discriminate the different TB spectrum. So until and analyze the assays to find how this spectrum, TB spectrum, can be identified by proper diagnostic tool or therapeutic intervention to handle all these three is very much needed. So the TB incidence, so WHO has some guidelines in order to reduce this TB incidence rate, number of TB related deaths, and also percentage of people with TB facing catastrophic cause. But where this is what is expected, but this is what is happening in the program. So now I'll just go into the science behind it and what we passionately work in the monocytes. So you should know that the Mycobacterium tuberculosis resides into the macrophage of your body. It is like a thief who is dwelling in the police station. So our body has a patrolling mechanism, which is the macrophages, but the successful pathogen goes into the macrophage and they survive there within the police station. So that is why it is very hard to eliminate the thief out of the police station. So this is a very interesting thing, like you should know. And we should also know how they are having advances to protect the host and what are the limitations which helps the pathogen to survive. So that is a very important thing. And for MTB, it is still innate, it is not completely understood. Yeah, so this is alveolar macrophage. What happens is an alveoli where what is happening when a TB bacilli gets into the system. So you should see it is transmitted by aerosol, air droplets. When somebody coughs or sneezes, it can go into a droplet and it is active up to 24 hours in the environment. So whenever it goes into a nasal droplet, it can go into the alveolar lungs. So this is the alveolar sac. So initial cells which comes into the contact is the macrophages. They pick up this bacilli, and then once they just phagocytized, they release some mediators, particularly the IL-6, IL-1B, and TNF-alpha. These are the pro-inflammatory cytokines which being initially released. And in addition, they also release some chemokines like CCL4, CXCL9, and CXCL10. These chemokines help in attracting the other components of the cells to the site of infection. So which is very essential. Then after this process is called chemotaxis, which brings about the T cells, neutrophils and monocytes to the site of infection. With the support of all these other cells, the MMP produced by alveolar epithelial cells, because some may escape from the monocytes and then go into the epithelial cells. So as a result, MMPs are produced. MMPs are metal-metalloproject proteins, and they come and then they also produce a lot of inflammatory reactions and MMPs. Together with the chemokines, MMPs and inflammatory mediators, they try to bring about inflammation. And this inflammation is mediated with the help of a majority of neutrophils in that place. Once all these systems is getting activated, the macrophage tries to digest the bacilli and to produce the antigenic component to the T cells. And it happens through the signal transduction here. Like you have this MHC peptides, where they take the soluble proteins and then structured proteins, cell wall proteins, are break down into peptides, and these peptides are being presented to the CD4. Similarly, the soluble proteins are also being excluded here. They are picked up by the dendritic cells, and these dendritic cells will have a downstream Th1 effector cell mechanism and induce the cytokine called interferon gamma. So this is very important to kill the intracellular parasite. Then not only Th1 and Th17 also gets into the system. Then together with, there is a small amount of affinity maturation or affinity binding of the protein, and based on the antigen antibody specific strength and it's called avidity, there will be some B cell process, and which in terms produce some plasmacytes. These plasmacytes, they give some matured antibodies, but in this case the cellular process is very important. The humoral response is very minimal. So this is very minimal. So the cellular component takes care of the majority part where the bacilli is being killed into the system. So this is what it happens into the alveolar sac and macrophages. But what is there is, in the monocytes, these monocytes basically differentiate into macrophages and dendritic cells. I think you all should have studied in immunology aspect about that. And within the monocytes, there are three different components. And these three different components are called the classical monocytes, intermediate monocytes, and non-classical monocytes. These monocytes are very responsible. The classical one is responsible for the phagocytosis. Whenever a foreign body comes, it has to engulf and take up the bacilli into it. The second one is intermediate monocytes, which is majority role is based on the antigen presentation. And the third one is non-classical monocyte. So these three are mainly identified by two different markers, CD16 and CD14. So when you have a system blood monocytes, you can have around this type of waterfall kind of system where you can classify the non-classical, classical and intermediate monocytes. And this intermediate monocyte is very plastic, heterogeneous. They immunomodulate a lot of biological functions. They help in the tissue homeostasis, cell differentiation, cytokine production and immune cell mechanisms. So this is the main function for the monocytes, and when this dysfunction of these monocytes happen, there is a problem in the handling the intracellular parasite. So you should know that they have the main role, and here in the case of MTB infection, it has an antimicrobial effect and also promotes bacterial survival. So this is the dual nature of the monocytes and macrophages, where we need to study exactly in order to curtail the mechanism. So how we can study the monocytes in TB, either by phenotypic method or by functional analysis. Functional analysis can be done in different ways, but here it is of mostly transcriptomal approach. And here, like these are all some of our earlier studies where we have seen some of the altered phenotypes, and some of them have different production of DNF alpha mechanism, and also these are all the other markers which are really helped in differentiating the monocytes phenotypically. And similarly, there is a prominent dysregulation of innate immune genes across the monocytes. So they are studied in PBMC level, not at a single cell level. And this Fc gamma receptor on signaling and type 1 interferon signaling is a major mechanism by which this functional attributes of the monocytes gets altered. And some of our own studies earlier, we have shown that this miRNAs and other mRNA mechanisms are completely different, where there is a different in these things helps in the heterogeneity of the monocyte, and they have a difference in the plasticity of the monocyte. According to that, the function of the monocytes gets altered. So I'll explain this monocyte study alone. So what are the major aims we have? So basically to functionally characterize the phenotypic subsets, and then to study the transcriptomic profiles of this monocyte subsets, and then the most promising candidate biomarker genes, and the network bypassing the transcriptomic profile will help in the differentiating the active TB patients to the healthy subjects. So this is what's needed in our field of research. So for which we need to have study subjects or the participants, which includes the spectrum of the TB disease, where they are the healthy controls, latent TB population, which is actually like our own population. 40% is latent TB, 60% will be healthy control. Then comes the drug-resistant or drug-sensitive bacillus, and then the drug-resistant cases. These are all the active TB forms who comes to the clinic with a lot of symptoms, and some can be managed with the treatment of 6 months, but the drug-resistant, they have to take at least for 12 months. So now we'll study how do you do the study, the phenotypic of this monocyte subsets. So this needs a very small instrument, which is a hematology analyzer, where the blood is being taken, and then from this, second thing is we also stain the cells with the antibodies, and we do it by FACS analysis. So when you have these two instruments, you can always study this particular phenotype studies. So here, if this is one of the picture where you see across the 4 categories, the numbers of the phenotype cells varies. So you can see in the healthy, latent, drug sensitive and drug resistant, the number of monocyte subsets are drastically increased. And this can be studied by means of MLR ratio, which is monocyte to lymphocyte ratio. So whenever you go to the hospital, they will ask you to take a blood test, and that blood test will tell you whether you have more monocyte or not. If you have more monocyte, you are infected, and particularly if it is intracellular bacilli, the MLR ratio is going to be high. So this is a very simple phenomenon which says that these monocytes are very high. Then elevate monocytes in the active TB. So among the monocytes, as I said, there are 3 different subsets. Among the 3 different subsets, which one is more and what conditions? So here we can see no difference in the classical monocytes, neither in the non-classical monocytes. Whereas in the intermediate monocytes, you can see there is a distinct difference in the number of monocyte subsets among the categories which are really active in disease. So this is being shown here, and this is a strategy. How do you do a gating of these monocytes in the FACS? Then the problem here is it is not eliminating monocytes alone. Other than monocytes, the dendritic cells also being reduced, because a proportion of monocyte is needed in order to control the bacilli. So most of the monocytes are converted into macrophages, but the number of dendritic cells are going to be very less, and it is being reduced. It is a very typical thing called as diminished DC subsets in active TB. And there are UMAP studies where we can see that the membranes have an altered expression of different markers, CD markers, and based on that we can see whether they are activated or not, and you can approximately find which cells of the monocytes are being activated. So that you can study by this methods. Similarly, the activation of dendritic cells, and there is a reduction in the dendritic cell subsets. The membrane of the disease goes down so that they don't sense the downstream effect of the immune response. So this is a summary of the phenotypes, where you can see there is in the active TB disease, the intermediate monocyte subsets are increased and they have been linked to the trained immunity. It is not specific for only TB. Any infection can have this altered immune response. And the dendritic cell population is decreased. So however, the differential frequencies of monocyte and disease subsets in the DR group are trivial and statistically not significant. The UMAP analysis reveals the different expression of monocyte activation and marked disease in the DRTB compared to the DSTB. So this is about the monocyte phenotypes, but there are some factors which determine the monocytes. It is not the monocytes, they change their phenotype. The environment of the body is a circulating phenomenon, soluble phenomenon, which helps the subsets to change and to alter. So for which the cytokines are very essential and chemokines are very essential. So we want to study what is the soluble factors which has a direct role in differentiating these monocyte subsets. So here, for which we need to have the blood and then plasma. Earlier we looked into the blood, now we are looking into the plasma, and then from the plasma you can do this Luminex assay and then we can study what are the things here. So this is the one thing, like we can check all the cytokines at the same level and see what are all the cytokines which are upregulated and where it helps the conversion of intermediate monocytes. So if you see that all these cytokine studies are upregulated except this IL-5. This IL-5 will be regulated only when you have a extracellular parasite like a nematode or a tapeworm or anything like that. If you have it, IL-5 is going to be induced so that they have a different mechanism to destroy the extracellular parasite. Otherwise, for all intracellular parasite, these cytokines are very essentials. So this is one another heat map where you can see how much the cytokines are being differentiated and how it is going to be differing among the 4 spectral conditions. So this, if you normalize to the normal healthy context, the latent TB will have these many cytokines expressed under these levels. When you have an active disease, the levels get increased, and when you go to the drug-resistant conditions, this goes multifolded. And this condition is called hypercytokinemia, where all the cytokines are expressed in a long-term process. And based on these cytokines, we can clearly discriminate the conditions like healthy to latent, latent to DS, and DS to DR. Predominantly, it is all IL-17 driven mechanism. These are all the top 8 cytokines which can cleanly identify all the 4 stages of TB. This is for stage specific. So this is a summary of the cytokines. Similarly, we did it in the chemokines also, where you can see there is an upregulation of these chemokines and there is no difference in the limited 3 chemokines. Similarly, when you want to see, there is a increase in this chemokines from drug sensitive to drug resistant. These are all the parameters of chemokines which helps to build the inflammation across the TB disease. This is again the markers where you have this. The potent chemokines are CXCL9 and CXCL10, which can be a marker for the disease. Now we want to go for the transcriptomic approach to study what is exactly the functions of the cells across the TB spectrum, and for which we need to do the RNA sequencing, study the miRNA and mRNA, and also there are other forms of RNA which can be decoded, and we can study the pathways of genes, how they are activated and where they are contributing to the functional dysfunction. So for which it is basically what we have done is, like we have not taken the PBMCs as such. We try to sort the monocytes, and these monocytes are sorted with the help of FACS, where they are directly sorted into the cells, and these sorted cells are further isolated for RNA, and they were doing by the next generation sequencing. We do the Illumina RNA technology. And there is another technology which is called nanostring, where it takes the biomolecules directly and read it. And this is going to help you how much miRNA is there in the body, and this can be enumerated. And based on the number of reads and RNA sequencing, we do a differential expression analysis, target prediction, and also study the pathway analysis. So this is a very basic step which can be applied across all the diseases and also all the models of techniques for transcriptomics. So here, the multiple genes have been differentially expressed across the countries, across the categories of the TB patients. And you can see how many genes of them are being downregulated, upregulated in the active TB as well as in the DRTB. So there is a rise of monocyte genes in the active disease TB compared to the latent TB healthy individuals. So during the transformation of latent TB to active TB, all these monocyte genes are upregulated. And here also, like, what you have to see is what are the pathways and mechanisms. Now there are AI tools are available. These AI tools will automatically identify the genes within the monocytes, and they can take which are all the genes which are really activated. So based on application of AI in artificial intelligence in the immunology, we have identified these are the different subsets which influence the interferon, inflammation, leukocyte activation, cell cycle mechanism. So these are all the pathways which is entirely altered. And they are being showing that in the healthy conditions, you see more of green dots, which means they are not activated. Whereas when you go to the latent stage, followed by active and DS and DR, everything is being activated. And this is bidirectional in approach. So which is a clear indicator of which gene can be actively used as a biomarker. These are all the pathways, how these monocytes help in the regulatory mechanism. And this is how a biomarker discovery is made. So we need to do a couple of approach where you can have this biomarker discovery. There is a future selection method and then stem pattern method. From this we have to validate the identified gene across the gene sets, and you have to see an AOROC. From there we can see whether it is responsive for the treatment or not. Then based on the treatment response we can say whether it is a suitable candidate biomarker or not. So these are the future selection targets where so many genes have been identified in categorizing the different TB markers. Similarly, these are all the stem pattern. So we have these 4 groups, and within the 4 groups, how they are behaving. Latent, healthy to latent, latent to active, and active to DR and DS. So you see some pattern are going down, some patterns go up, and some they drastically reduce. So based on these following patterns, which are totally in silico and bioinformatic approach, you can identify the genes which are really categorized, and then we can help them to promote as a biomarker. These are all again validated mRNA candidates, and these are the genes which are being really predicted by artificial intelligence. And these are the genes which are really predicted by the treatment response genes. So based on these identified ones, we have identified what are all the possible mechanism, how the monocyte which functions during the stages of disease. And these mRNAs, they have both diagnostic value as well as therapeutic value. So unless and until you identify these things, you cannot go beyond the research where you need to contribute what are all the genes are being activated and how they can be controlled. The host-directed therapy is entirely based on this understanding. So unless we study these genes, a host-directed therapy cannot be applied in order to handle the bacteria. You cannot always depend on the antibiotics. You know that from the last session, you know that antibiotics lead to antibiotic resistance, where we need to add on the number of drugs. So for TB right now we have 13 to 14 antibiotics. If you are drug sensitive, you may have to have 4. If you are drug resistant, you have to take 12 Plus, depending on the issues, you have to add more number of antibiotics, which is going to complicate the treatment plan, and it is not an advisable thing. So the recent research is, how do we target the host to take care of the monocyte dysfunction? As a result, the survival of the pathogen can be eliminated. So that is why our research is focusing on these kind of approach, and we are attempting to address some of the questions. So as we did mRNA, we also did miRNA. So what is the difference between mRNA and miRNA? Any idea, students? Why do you need to study miRNA here? Anyone? Okay. See, the mRNA is basically a conversion of DNA to mRNA and mRNA to protein, which is induced in the translational mechanism. Whereas this miRNA is a regulatory mechanism. This miRNA has a potency to go and bind on the transcription initiation site of mRNA. When you have an upregulated mRNA, it will never allow the certain mRNAs to express. As a result, the proteins are not being decoded or being produced. So this is very tricky in terms of regulatory phenomenon. So that's why we want to study the miRNA also. So this can be studied, and mRNA has a regulatory mechanism across all these pathways: phagocytosis, apoptosis, autophagy, inflammation, cytokine differentiation, NF-kappa B pathway, and Taylor mechanism. So everywhere it is being regulated by miRNAs. And how these are studied, you can study either by having human studies, or by studying murine studies, or by cell lines. So here what happens is, as the disease progresses, there is a down-regulation of miRNA. So in a normal condition, you have multiple miRNAs. When a disease starts, the miRNA levels get reduced. As a result, what happens is, there is no regulatory approval for all the chemokines and cytokines. So as a result, the chemokines and cytokines, they are getting upregulated and create a condition called hyperinflammatory conditions. This is mainly because the levels of miRNA are down-regulated in the DS and DR conditions. So these are all the fold changes, how it varies and how it differentiates across the disease patterns. So these are all the differential miRNAs which are exclusively studied across the stages or the spectrum of the TB disease. And they also have a bidirectional response. That is, it is upregulated in the normal state, downregulated in the disease state. So it will be easy to correlate how to do and control the disease stage activation. And this is the summary. And I just conclude by saying a small summary what it happens in a normal stage and in a disease stage. So you can see in a latent TB infection, where it is happening in all of us, at least 40% of our people, there is a slight increase in the monocytic mRNA. There is a cytokines and chemokines, it gets elevated. The monocyte mRNA gets reduced. The monocytes, intermediate monocytes and lymphocytes and dendritic cells, they do not alter. As a result, the suppression of multiple immune responses takes place at this stage. That is why we are all protective for TB. Whereas if you go to the active disease, monocytes again upregulate, intermediate monocytes particularly, it goes up. Monocyte mRNA gets elevated, cytokines and chemokines, they get elevated, whereas the monocyte mRNA, lymphocyte and dendritic cells, they get downregulated. As a result, activation of multiple immune responses are being elevated. So all this study was made possible, and this is one of my students' Ph.D. work. So I should thank her for doing this work. It is Pavitra here, and Clinic Division Statistics. And this was funded from NIH USA, and Tamil Nadu State Government, nanostring is from these people, and also the entire funding was given by DBT. So I should thank everyone for this possible thing, and I should thank you for patience.
session is open for queries.
Yeah, I'll be happy to have some questions. Need not worry whether it is a basic or advanced. Feel free to ask. If I can answer your questions, I'll be happy to let you know. So if there is no questions, means it's either two-way. Either you understand everything or you don't understand anything. Yes. So you tell me frankly, if you don't understand anything, then I can try to make you understand something. So this is an interactive session, and it's very easy to learn something. We are working from those researchers who directly work in it. Yes, ma'am.
Sir, the presentation was very nice, sir. Thank you for the interesting presentation.
Thank you.
I have one question, sir. Why you specially focused on monocytes and their mRNAs likewise?
Yeah. So as I said in the initial talk, this TB goes and resides within the macrophages. This monocyte is a precursor cell of macrophages. Until and unless this cell differentiates into macrophage and the macrophage is not functioning properly, the bacilli can survive into the macrophages. So rather than studying all other cells, we have to focus only on the cell which is inhibiting or it is having a habitat for Mycobacterium tuberculosis. That is why we want to study the role of monocytes, which in turn converts into a macrophages. That is very essential. Yeah.
Thank you, sir.
Thank you.
Good afternoon, sir. So, in the period of COVID-19, we used to say, basically in South Indian people used to say because of Covaxin or COVID Shield or Sputnik, whatever it may be. But because of this English medicine, people used to get suffer of side effects, even though nowadays also they have been saying like that. Is there any traditional medicine system which used to cure any TB kind of things? Because in COVID-19, South Indian people used to say like that, Kabasara Kudineer is more efficient, more effective for used to cure coronavirus. Is there any Tamil or Indian traditional medicine used to cure TB kind of disease, sir?
Yeah. So there are multiple medicines, traditional medicine as well as herbal medicine. One, if you ask the Siddha researchers, they will tell it's called imbural. Imbural no oru chedi irukku. And the chediyoda extract is helpful in modulating the macrophage functions. So that is there in the Siddha research. And all we have to do is, we need to find a mechanism where this immunomodulatory activity can be enhanced so that the immune system gets expressed. So if you can activate the immune system, the pathogens can be very well maintained. So if at all we can intervene in the immune system for immunomodulatory thing, you can always upregulate. One particular thing is vitamin D mechanism. Vitamin D has a direct role in stimulating the antimicrobial peptides along with the modulatory activity of monocytes. So these are all targeting the host and they don't target the bacilli. That is why it is called host directed target therapy, which is very helpful in handling the pathogens.
Thank you, sir. And somewhat people used to say like that in this era or a future era comes all about artificial intelligence. Is there any particular tool or a bioinformatics tool used to cure infectious disease like mycobacterium?
For cure, we don't have any alternate agent, but for diagnosis, we have it. So basically, we take an X-ray and then we put it into the artificial intelligence and machine learning. So based on the X-ray, you need not have a radiologist to say who is being infected by TB or not. So nowadays, we are developing a tool, and we can easily say by giving a direct X-ray, it can say whether you have a TB infection or not. So that it is more helpful in the diagnosis rather than the treatment.
Thank you.
Thank you.
Any other queries? Thank you, sir. We now request Dr. N. Vidya, Professor, Department of Microbiology, to present shield to our keynote speaker as a token of appreciation. Thank you, ma'am.
A moment of gratitude makes a difference in your attitude. We would like to invite Ms. Shruti from First BSc Microbiology to deliver the vote of thanks.
The essence of all beautiful art is gratitude. Now, I like to take this opportunity to express my heartfelt gratitude to Dr. B. Ramalingam for his informative lecture on monocyte heterogeneity in pulmonary tuberculosis. Thank you, sir, for your invaluable information we got about this topic effectively. And I would like to thank this session's chair, Dr. P. Arun, for introducing the guest. Thank you, sir, for your valuable presence.
Let's move on to the technical session 6, which delves into the concept of food pathobiome, food resistant, the big future. We now request the keynote speaker and session chair to occupy the dais.
In knowledge pays the best interest. We now invite Ms. T. Legashri from First BSc Microbiology to give an introduction about Dr. M. Pungodai, Professor, Department of Biotechnology.
Good afternoon, everyone. I am highly honored in welcoming Dr. M. Pungodai, Associate Professor, Department of Biotechnology, who has 10 plus years of teaching experience and 5 years of research experience. She is highly specialized in the area of plant pathology, molecular biology, and mushroom bioprospecting. We are happy to have you as a session chair for this technical session, ma'am. Dr. M. Pungodai had completed her BSc graduation in the field of biotechnology and biochemistry at Avinashalingam University, MSc in biotechnology at Bharatiyar University, and her Ph.D. in biotechnology in Sugarcane Breeding Institute, Coimbatore. She had her vibrant participation in seminars, conferences, workshops, research publications, book chapter publications, and faculty development programs. Dr. M. Pungodai was renowned as proficiency holder in biotechnology. She had received Sri Nalla G Research and Innovation Award for her best paperwork and as best professor in research contributions. Ma'am, with her outstanding initiatives, had earned grants for 3 of her lab-on projects. With all the milestones she reached and to be reached, I feel a great pride in welcoming Dr. M. Pungodai to introduce the guest speaker of today's session.
Good morning, one and all. I feel very much privileged to introduce the guest of honor before you, Dr. S. Vignesh Sir, Associate Professor, Department of Academics and Human Resource Development from National Institute of Food Technology, Entrepreneurship and Management is before us. And Sir has completed his graduation and doctoral studies in Bharatdasan University. Sir's experience is more than 12 years. Sir has renowned interest in research, which is evident from n number of funds that Sir has in his hand. So Sir has several projects which is funded by Sir. He was a principal investigator for food waste to microbial polysaccharides by fed batch fermentation in the year of December 2021 to 23, and to the latest Sir has received several funds on industrial projects too for development of different flavored humus, juju based beverages, ghee processing are a few which I want to say. And apart from these projects, Sir has also handled several other projects. Sir has more than 80 publications, book chapters, and H-index and I-10 index is evident that Sir is a renowned researcher, and we wish to hear more from Sir, and I ask the students to know more from Sir's subject expert. Thank you. I welcome you, Sir.
Thank you, ma'am. We request our session chair, Dr. M. Pungodai, to honor our keynote speaker, Dr. S. Vignesh, with a shawl and memento. Thank you, ma'am. Sir, the dais is yours.
Shall I operate, sir? So, respected principal in absentia, and my fellow keynote speaker, sir, my good friend, Dr. N.G.P, and dear colleagues and my dear students, a very good afternoon to all. Dr. N.G.P have given me only 45 to 50 minutes time to explain my topic. I know very well it is a time for lunch and you people may be starved in this condition. So trust me, I will keep my promise. I will complete my lecture within 45 to 50 minutes. I have a watch, okay, no problem. I will complete on time. Trust me, I will keep my promise. And the second thing, today's world, you people know, and you people are chatting with yourself, and this side also I'm seeing, when you see the Batman Begin movie, the heroine said to the hero, like he asked her, what is you are talking? He said, just a girl thing. It's just a girl thing. That's what I understand. Some of the girls are speaking anyway. I understand. That is most important. You think, right? So let me tell you something about food. Actually, every day we are eating the food. Once upon a time, it was when our ancestors had some of the food. That time that was not lavish. For their survival, they had food, and frequently they couldn't get the proper food. And at that time, they have a nutritious food. But nowadays, food is lavish. And if you want the food, we are ready to spend a lot of money to get the food. But after getting the food, are we taking a good food? I'm just asking you a question. Are we taking a good food? That is the biggest question. As you know about this genesis, see, we people are made up of, the 6 compounds: carbon, nitrogen, oxygen, hydrogen, sulfur, and phosphorus. This is the major component. Major and minor compounds are also there. But
we people are totally depend on these are all the genesis. Out of this 5 genesis, 2 genesis are very important. Once upon a time our ancestors said, Unavum neerum virpanaikalla, food and water is not for sale, but this is the biggest market in the world. Globally, this is the biggest economical value, and many people are having this kind of business nowadays, right? And my question is, are we eating a good food? We totally depend on 2 things apart from the air. We are surviving the land and we totally depend on the water. We are having only 6% of fresh water, less than 1%. 6% is okay for managing the things. But are we getting a good without contaminated food? That is the biggest question. In Kambaramayana, he said one important poem, okay, in the Balagandam. Nadiyin pillai என்று நறும்புணலின்மை அற்றே பதியின் பிள்ளை என்று பயந்து நம்மை புறந்தான் மதியின் பிள்ளை என்று மகன் பிள்ளை என்று மைந்த விதியின் பிள்ளை நீ எதற்கென்னை வெகுண்டதென்றான். So for this session, it is not closely related to this poem. Okay, the situation was Rama came to the forest along with the Sita and Lakshmana. The Bharata followed him, so that the Lakshmana got angry on Bharata, so he wanted to fight with him. That time Rama said all this story. I don't want to explain, but the first sentence is very important. Nadiyin pillai endru narumpunalinmai. Kamban said the river is not responsible for its pollution. Nadila nalla thanni odadadarku nadi karanamalla. So we can proud of it. Our ancestors also polluted our environment. 800 years back, our ancestors have polluted our environment. They are also involved in this kind of activity. What kind of attitude we have nowadays? We thought the river reservoirs or the aquatic environments are ultimate sink for all our byproducts and waste. They are also did the same kind of thing, but the only difference, that time they respect the nature and the nature has the self-purification capacity. But nowadays, it was reduced. The nature is losing the self-purification capacity, and we people are completely polluting our environment. In the roadside, in the highway, if you are traveling in the highway, Indian transport system, you can see at least 50 years or 10 kilometers or 20 kilometers once, they displayed one board. Please avoid open defecation. Don't split it on the public place. Avoid the urination in public. All these slides you can see. As a microbiologist, I am always telling to my students, the microbiologist and monks are same. Monks means Munivargal. These 2 fellows are always doing research on the inevitable and invisible matters. Right, and they only know the real truth. Okay, you can't see the microorganism, but you can cultivate it. That is responsible for all kind of your activities. That is the microbiome. And this is the point. In the roadside, you can see the pictures, and they mentioned open defecation you have to avoid. As you know, as a microbiologist, you know very well the composition of your fecal matter. 5 to 25% of your fecal matter composed by the enteric microorganism. Your enteric microorganism composed of commensal organism, pathogenic organism, as well as beneficial microorganism. And this much amount, I'm just telling you one brief example. Your Rangaramanujam sir tell me that, sir, give some case study related to students, what you have experienced, so that it will be useful to their day-to-day life. Okay, so I'm telling you one interesting story. In your esteemed institute, approximately I hope the 10,000 students are here, studying here. How much amount of urine and fecal matter every day you are releasing? Average, and an average is 30 to 50 ml of urine, minimum, and an average human being. And the fecal matter is approximately 200 to 300 gram. 1 gram of your fecal material contain almost 10 power 7 to 10 power 9 microorganism. You know 10 power 7 to 10 power 9. That is approximately 1 gram of your fecal matter composed of 1 crore of microorganism. 1 crore microorganism. That much amount of microorganism will be settled in the septic tank, and they have frequently collected the septic tank. Just imagine the Coimbatore district, just imagine the whole world. How much amount of waste every day we have created, and where they, after they have collected this waste, where they have put it. That is the first question. And second thing, after this Vastu will be boomed in our area, they said in the Isanya Mulai, that is in the northeast corner, we have to put a borewell. For your neighbor, that is not a northeast, that is the northwest. Normally the septic tank will be placed on the northwest. For you, that is the borewell water. For your neighbor, near to the borewell water, they will place the septic tank. All right. So the groundwater will be contaminated as per the Pollution Control Board of Tamil Nadu. You just see, and the government data is also available. You just see, most of our aquatic environments are completely polluted because of all these problems. Not only this fecal matter, not only this matter, completely because of antibiotics, because of pesticides. In the year of 1999, Hong Kong scientists, they have published one data that was stunned the whole world. The women, they have collected a sample from the women's. They have given their breast sample. So far, we believe that in the world, purest one is our breast milk. Okay. So they have collected a sample from the different women. I have a wife, I have a daughter, I have a mother. Right. Like that, in each and every country, the people, those who are residing to the agricultural land, right, they are ready to give the sample. They have tested the sample and they find out that sample contain high amount of OCP and OPP values. OCP, organochloropesticide, and organophosphopesticide. After that only, many countries have reduced the usage of pesticides. You know how many of you non-vegetarian? Just to raise your hands. இல்லை கொஞ்சம் தூக்குங்க, ஏன்னா மதிய நேரம் கொஞ்சம் நானும் கொஞ்சம் ஆக்டிவாக இருப்பேன். ஃபைன், தேங்க்யூ. Right. I will show you some of the report. After that, you have to decide whether you can able to eat the meat or not. Okay. Aquaculture environment especially, I have, when I was doing the Ph.D, I have traveled throughout the Tamil Nadu coast as well as the Andhra Pradesh coast and Kerala coast. I have traveled more than, out of 7,000 kilometers, I have traveled more than 4,000 kilometers for the sample collection. Many of the places, yes, excuse me. Yeah, please, thank you. So many of the places I have seen, the aquaculture, they have cultivated the prawn and the fishes without any proper permission, without getting the doctor consultant. They are using umpteen number of antibiotics into that. Okay, they are approaching some of the pharmaceutical company, and illegally they have collected lot of the immature antibiotic in the sense that it is a kind of precursor. They have collected antibiotic from them and they will put it in the aquaculture. What will happen? We call it as a bioaugmentation. It will be accumulated in the fish or prawn body and we will consume it. Okay, that is what happening. That's what we said. Food hazard and water hazard is closely related. I have taken this picture. If you see the coastal environment, this is our scenario. One side, this is the point source pollution, just to see that yellow color. Non-point source pollution, not a point source. A point source, you know, the sewage. Non-point source, this much amount of waste will be deposited. Just to take an example of our Chennai beach, that is the world's second largest beach, right? Approximately in the weekend, I hope nearly 5 lakh people will be coming to that place and enjoying. Out of 5 lakh people, just 10% is children. Just 10% I am taking, 50,000. 50,000 young children are involving in the open defecation process and open urination process. Just imagine the load of, when you read the, when you see the Troy movie or when you read the Homer's Iliad, he said, the Achilles will going to take a beach of Troy with the 50 men. He said, he is having only 50 warriors, but he is going to take a beach of Troy. There are 10,000 warriors are there. He is just going to take a beach of Troy with 50 men, he said. Like that, every day, every weekend, each and this much amount, right, each and every human being is contributed this much amount of pollution to our aquatic and food ecosystem, right, food system. And this is the halobiont disease, a very important one. You are not only, you are the only one person, right, okay. You are surrounded with all this kind of environment. So the halobiont is nothing but it is a assemblage of all these things, antagonistic microorganism, mutualistic microorganism. So the microorganism is nothing but when compared to your cell, the organism which is present in your body is very high. That much trillions and trillions of organisms are there. That is made up of different genes. Some of the genes are responsible for antibiotic resistance. Some of the genes are responsible for pesticide resistance. So that that gene can be easily transmitted from one place to other place, or variable transformation process or transduction process or conjugation process. It will be transferred from one organism to other organism. It will be easily carrying the antibiotic resistant genes. We call it as a ARG. This is a news bit. Almost frequently you can see all these things, and we people are believe that apart from land and water, right, we people are believe that we are inhaling the fresh air. See this paper, Global Survey of Antibiotic Resistant Gene in Air. Globally they have collected 10 sample, one sample in Chennai near to the beach side, back side of the presidency. They have collected this kind of sample. They observed antibiotic resistant genes are present in the air. This is the current scenario, right? And very well about microbiome, and we call it as a terminology foodomics. Apart from the proteomics, metabolomics and genomics, all these things, the foodomics are very, very, very important, because almost it is a completely nexus on the water and food. So that's why we call it as an enteric bacterial contamination. Especially when we are talking about the quality of water and food, we are talking about the enteric organisms, because the enteric organism can easily enter into that system through that urine or fecal samples. So these food systems are almost contaminated. Even for my case study, I just show you later the slides. So once upon a time we believed that the food safety means the food should not contain any harmful matter. But this is the definition of WHO. You can clearly see this one. The absence or acceptable or safe level of contaminant, adulterant, naturally occurring toxins can be present in the food, but that should not cause any harm to the human being or the consumers. This is the current scenario. So especially during the COVID time, people are they run behind the jewel or money, right? They ran behind the grocery shop and they have buy lot of food material. Mostly that is a packed food material. 10,000 rupees the refrigerator, ordinary refrigerator cost. 80% of this country, that money is not affordable. They are not they are not ready to spend that much money to buy a refrigerator. So they are prefer only the packed food material. So knowingly the adulterant may be present. The preservatives are may be present. Sodium benzoate or potassium benzoate, a lot of sorbic acid, citric acid, acetic acid, many preservatives we are using. But those preservatives should not affect the human being. But if you are taking as a food scientist, food technology institute, right, one among the premier institute in the food technology in this country, I am telling you, if you are taking frequently this kind of packed food material, there are chances to affect your organelles, right? So we call it as a 4 things: clean, separate, cook and chill. 3C, 1S we call it as. We have to adequately, we have to cook, we have to properly clean it. Separately, we have to keep our food in the refrigerator, right? Why we have to study about this food safety? What is the connection with the food pathobiome and the food resistome? This is the important thing. Worldwide, more than 4 lakh people died because of food contamination. Our contribution is more than 1 lakh. So 1 in 4 member Indian, 1 in 4 member, we people are die because of food safety. So this is a very important thing. So my question is, the irony is, are we eating a good food? The food may be cooked by your mom or your wife or your spouse or anybody, this Vikram Tata or your mom or this kind of a thing, right? Are we eating a good food? Can you claim, sir, I am eating a sterile food this morning, sir? I'm not complaining your hostel or your institute. I'm asking, right, your mom is preparing or your wife, husband is preparing, right, whoever prepare. Are we eating a sterile food? No. No, not at all. Not at all possible. As a microbiologist, you know, whenever we are coughing or whenever we are talking, some kind of microorganism will come from your mouth. Some of the heroines are come to the toothpaste advertisement that they said you have to use our toothpaste. It will provide 24 hours protection. Nothing happened. Not even 1 hour it will protect. After you will wake up in the morning, you can just rotate with the using of your tongue, you just rotate your teeth. You can see, you can feel some of the biofilm will form on your teeth. That is the microorganism. Especially Streptococcus mutant and Candida albicans is the main organism. So even when you are writing an examination, if you commonly close your mouth, after 3 hours just to open your mouth, some smell is coming. That is because of microorganism, right? So it is closely depend on your health system, how much amount of organism you are taking, engulfing in your body or inhaling in your body. Out of that, how many organisms are pathogen? And then third point is your immunity. Based on these 3 criteria, the disease will come. Okay. And what we have to do? This is the important thing. The chain-like process, we call it as we have to follow all the practices. As a microbiologist, you know the typhoid Mary case. Typhoid Mary, right? She is a pervert. She worked as a waitress in the Paris hotel and she served the food to the customer. While she is serving the food, she have mixed her own fecal matter with the food. She did it. And normally, begin the screen, what kind of food they are providing. While the waiter is bringing the food, if he cough while he is bringing the food, will you eat that food? But begin the screen, what is happening? Right, okay. So we have to follow all these things, good manufacturing, good agricultural practices, and very importantly, good housekeeping practices. These are all the practices we have to follow. And this is the real case study of microbiome, pathobiome, and resistobiome, which is closely related to food. And this is the scenario. So this is the latest information. One side, India is drowning in its own excreta. That's what I said. This is a Hindu newspaper. Namalude pollution nale India moolgiit irukku solrom. The coastal side it is very common. The other side all these microorganisms and the genes are spreading a lot. Okay, and this is that's what already I said. The 100 trillions of microorganisms, approximately the weight is around 200 gram. That play a very important role in our body. I tell you one example. The colostrum, that is a seam ball nu solluvom. After the baby come to the earth crust, just born baby, 24 hours or 15 hours or 2 hours, immediately the doctor insists to provide milk, insists the mother to provide the milk to the baby. Or if the mother does not provide, means the milk is not secreting in the mother's body, immediately they go for an infant formula. How it will be digested in the infant body, how it will be digested in the infant stomach, that is related to microorganism, that is a LAB and the bifido organism. Okay, the colostrum is also a complex molecule. We call it as a HMO, human milk oligosaccharide. It is a complex molecule. It is a just born baby. It in the earth crust, just 24 hours is there. After 24 hours, we are giving the milk, or immediately we are giving the milk. So it will digest in the baby's body. So it will be easily digested because of the organism. But what is really happening in the inside of the stomach? Before that, you should know about the food microbiome, pathobiome, and resistome. The food pathobiome is nothing but the pathogens which is present in the microbial community of pathogens which is present in the food. And the resistome is a resistant gene which will be transferred from pathogen to non-pathogen, as well as some of the genes there is a chances it may be transferred from non-pathogen to pathogen. And this is the reality. The food chain, it will be completely polluted nowadays, and it's in our hand. We have to restore it. And this is the case study I have done. The study, we have published several papers in this. I'm just giving the glimpses. Okay, we have taken the food, right? The food is normally provided in the market. Lot of foods we have taken. We have taken more than 22 food. We have published this paper in Food Science and Biotechnology Journal, as well as Environmental Monitoring and Assessment Journal, and one more journal, International Journal of Food Technology, and some of the papers are in the pipeline. So you may refer it, I will share it with your faculty members to create awareness. We have taken the food as well as we are taking the packaging materials. What kind of, normally people are using different kind of packaging material, right? We put it in the food for 15 to 20 minutes. We have taken the food after taking, we just keep it for outside 2 hours. After that, we have collected those samples for the analysis. Okay, and we have find out some of the biofilm organism. We have find out in that biofilm we have seen umpteen number of organisms present in that. And lot of organisms are pathogens, and we don't know whether it is pathogen or not. It belonging to that group. This is the plate my students have done it. These are all the studies we have done it. Even we have studied the MR by using the MRS agar. We have studied the lactobacillus, and this one you have to see. Like we have done the ESBL study, extended spectrum of beta lactamase. You know very well, right? It is in the another step of ABR, and we have done the preliminary study and the secondary double disc study, and advanced improved ESBL kit method also we have tried, and we have observed out of 100% of organism, nearly 40% of organisms are producing the ESBL. Out of 100, whatever I said in the previous one, not all the microorganisms. I have chosen the organism which is belonging to the genus of the common pathogenic genus. Okay, right. So most of the organisms are resistant to, most of the organisms are secreting the ESBL, and at least 70% of the organisms are resistant to one antibiotic. Once upon a time, people are directly, if you go to the hospital, they will pierce the needle and inject the antibiotic. Now they will checking whether your body will accept that antibiotic or not. Then only they will inject it. Okay. So this is another study, same kind of studies. And this is the photograph we have taken for SEM and CLSM image. We wonder, after we will store it, we keep our plastic material, packaging material inside the food, not a food waste, not any other material. After that, we have observed under the SEM and the CLSM, this much amount of organism we have observed. And we have used some method to protect it, and the later stage H sample you have seen, the H1, there is no microorganism. Almost 80% of the microorganism controlled by whatever treatment we have provided. We have used some material to control it. This was happening for active packaging we are developing. We are doing this one, right? And this is a scenario already I said, the Vellore CMC report. If they are producing 1 kg of beef, they are using 45 milligram of antimicrobial. If they are producing 1 kg of chicken, they are using 148 milligram of antimicrobials. If they are producing 1 kg of pork, they are using 172 milligram of antibiotic. Almost knowingly or unknowingly, right, we people are taking antibiotic. Sorry to say this, some of the ladies are here, many girls are here, even including I'm telling to my mom and my wife. So without doctor prescription, almost you have some pills in your handbag. Am I right or not? Whatever problem comes, we are not approaching the physician. Directly we go to the pharmacy, right? We ask some guy, we ask the guy who is standing there. We don't know whether he have efficient or not, whether he have completed the B Pharm or M Pharm or not. Even some 10th standard student will be there, and he suggests you like you can take this pill, sir. Same combination, whatever you ask, this is not one. We have this one, you can try this one. We have taken. I'm just telling you, when I was doing my postdoctorate in South Korea, I was affected by severe fever and cold. I approached the pharmacy. He said, I will not give you any medicine, sir, just contact doctor. Approach a physician. After getting the prescription, only you just come to my pharmacy, I will give you the medicine. This is the condition of most of the world, but it is not a condition in our country, kind of developing countries. We have to change it. And this is the report, you have to see this one. If they have collected a sample from different chicken, especially near to the Delhi region, this much amount of antibiotics present in the chicken. So this is the real scenario. What is the regulation of FSSAI? The FSSAI said that this much amount may be a tolerance limit. Here you can see the mg per kg for fat, for cattle, muscle, kidney, liver, even in milk, 0.05. But we don't know in animal husbandry how much amount of antibiotics they are using. with the doctor prescription or without doctor prescription, right? How much amount of antibiotics they are in the real world they are using, we don't know. So we should create awareness. If you are animal maintenance or if you have animal husbandry, you should not use it. Get a proper permission or get an opinion from the doctor, then you can use it. And this is one report I want to show you how this will be evolving in your body, the microorganisms, right? After you will birth maybe a cesarean or the normal delivery, if the baby coming along through the vaginal tract, it not only carrying the placenta, it also carrying some of the microorganism which is present inside the mothers, or as well as the microorganism which is present in the vaginal tract, the baby also carrying all these things. If it is a cesarean delivery, the persons who are all handling the baby, from them the microorganism will transfer to them and it will be adapted. Okay, but this is the case in 1 year children's. In the feces matter, the feces they have collected from the 1 year children, and they have find out a lot of antibiotic resistant genes are present in the feces. You know very well it is come from the intestinal tract. So that much amount of antibiotic resistant organism present in the enteric region, and it will be released along with the fecal matter. And this is also one of the important MDPI review paper, right? Antibiotic resistant in pediatric infection. It is we call it as a co-infection. As a microbiologist, you know, this is also a problem. And here you can see the gut microbiome. The gut microbiome is very, very, very important. That's what I said. The HMO will be easily denatured by the microorganism, beneficial microorganism, lactobacillus and bifidobacteria. And the mother, from the mother milk, they will also get some amount of immune boosters, right? This will be affected by this kind of antibiotic resistant and antibiotics. If you are frequently taking antibiotic, you may be affected by dysbiosis. Dysbiosis means improperly your beneficial microorganism present in your stomach. That's what our ancestors said, whatever you are taking, whatever food you are taking, but at least in the lunch time you have to take some amount of buttermilk. Because it contains a high amount of probiotic, it will protect your intestinal tract, right? And this is the way, you know, right, the organism, you are a microbiology student, I don't want to explain it again. It can be easily transferred from one organism to other organism. And this is the situation. Now, the one side we are facing the pathogenic microorganism, and we totally depend on the beneficial microorganism. The problem here is the beneficial microorganism can get the antibiotic resistant genes from the other organism. It will become resistant to antibiotics. And one more problem, and sometimes there is a chances, same kind of gene, some of the toxin producing gene may be transferred from one organism to LAB organism. There is a chances to produce a toxin from the LAB organism. This is another problem. We have taken this kind of study, and we have developed non-ABR lactic acid cultures, mass culture, en-mass production of starter culture by using the food waste as a medium. But when we have studied this kind of LAB related ABR study, we wonder mostly around 30% of LAB organism containing the antibiotic resistant gene. Right. And this is the problem, already I said, the gut microbiota, which is not getting them, which is also affected by the antibiotics. And this is the one thing, right, the single species, multi-species problem. And the host gut microbiome is totally, completely, affected. And it is easily receiving the antibiotic resistant genes. And how we have to mitigate this problem, right? And already I said, if no gut microbiome, there is a cross-linking between the connection point between the brain and stomach, or it is closely connected with this LAB organism. If you are not having the proper LAB, you may have all this problem, atopy, obesity, and autoimmune diseases, right? So sufficiently you have to get the probiotic as well as prebiotic. And the second thing, whatever cultures, by using the starter cultures, whatever matters they are producing, especially fermented food they are producing, 100% you can believe it, that is a gross status. After getting the gross status only, they have used on the starter culture. Gross status means generally recognized as safe. It should not produce any toxin. It can be easily survive in the eukaryotic cell. It will not affect anything. It can be tolerated in the low pH, high temperature, low temperature, all the tests undergone, then only they approved the starter cultures. So we are mostly we can depend on the starter culture, because NMAS level, it is very difficult to control antibiotic resistant problem. One side we have to reduce the usage of antibiotic. If it is necessary, we can use it. Once upon a time, people are using the first generation. Now fourth generation drugs we are using, right? And frequently we are tested in our body whether it is resistant or not. This is also an issue. And this is the real scenario in your gut. If you are taking any pills or anything, what is exactly happening in that? This side I am just giving the LAB, the role of LAB in your body. The complex molecule will be easily degraded by the LAB or bifidobacterium, and you will get the SCFA, short chain fatty acid. The other side you can see the unresolved compromised inflammation gut barrier. That will also affected by this kind of organisms, right? The gut will be there, the mucus and the mucous membrane. You can see some of the fecal matter of your infants, your maybe nephew or niece. The fecal matter coming along with some yellow color jelly like a material, sali mari irukum. That is the mucus. That is the first barrier. Then the organism is very virulent, then it will penetrate that and adhere on the mucous membrane. Then only it will cause the inflammation. So this organism involving in the block adhesion process, it will not allow those organisms to bind on it. But nowadays, if it is antibiotic resistant or if it is a toxin producing organism, then it will be the problem. So these organisms can able to cause the infection, right? And this is what I said, the block adhesion, right? This is an important one. And now we coming to the point. So food testing is another problem, and sampling is also very, very important. And maybe your teacher, your staff may ask you to collect the sample from one area, maybe Maradamala region. I know only this place. Maradamala region. Apple, he asked you to or she asked you to collect the sample. And your friend telling you like, hey, why you are collecting same apple is available here. We can collect the sample from here. My question is it right? Not ethically, just practically you just tell me. Is it right? Normally, how the sample will be collected, that is a separate portion. As per FSSAI, we have to collect. If you are collecting this much sample, finally we required, as per FSSAI, we required only 400 ml of liquid water for analysis, 100 gram of solid sample is analysis. I am the technical manager of microbiology in FSSAI referral laboratory, one among the 14 laboratory in the country. Right? The sampling is very, very important. I am telling you, this much amount of sample is required for testing. But we have collected this much amount of sample, this much lot. So this small quantity of sample represent the whole lot. Right? So, if you cross the Coimbatore, here you can see this is the N.G.P sample, college sample, that is a Marudamalai sample or Singanallur sample. If you cross the border, that is a Coimbatore sample. If you cross the border of Tamil Nadu, that is a Tamil Nadu sample. If you cross the border of India, that is a India sample. They may collect a sample from Assam. Nowhere we are connected with Assam, but that is called it as an Indian sample. So the sampling, aggregative sample is very, very important in the incremental and aggregative sample. After you will collect the same quantity of sample, you have to aggregate it, homogenize it. From that you have to take a sample for the analysis. Okay, just for your information I am telling. And this is the microbiological analysis advanced equipments now we are using. Right, within the matter of 1 or 2 days, if you have a VIDAS or VITEC or sequencer, this is sometimes the food will come to the port. Within the matter of 3 days, 72 hours or 42 hours, we have to clear the sample. In that case, we have to totally depend on this kind of advanced equipment. Immediately we have to find out the organism. In this case, 4 terminologies, I hope your teachers already told you: isolation, identification, detection and enumeration. These 4 terminologies are very, very important. As per the FSSAI, enumeration is second. The first one is detection. Whether any pathogenic microbe or particular microbe is present or not. In that case, we are following completely this VIDAS or VITEC kind of equipment for the analysis. And as per the FSSAI, we are following only IS method. In practical, your teachers told you, like I am also telling to my student, for bacterial plates you have to incubate it only at a incubator 37 degrees Celsius. But as per the FSSAI, if you are following the IS method, we have to incubate the plate at 30 degrees Celsius. for TPC count, total plate count or total viable count or aerobic plate count method. Then we have to leave it for 48 to 72 hours. This is the way we have to analyze it. When you go to any country, they will follow only IS method. Most of the NABL laboratories, accredited laboratories are following only IS method. Whenever you are doing this kind of research, if you are maintaining 20 degree or 40 degree or 50 degree, whatever, you have to just mention in your article. That is mandatory. Okay. When you just go through the FSSAI standard and guidelines, this is very important, right? They have mentioned how much amount of preservatives you can use it, how much amount of antioxidant you can use it, and how much quantity you have to use it. Everything they will mention. Not only this kind of preservative, and this is the microbial load. And I'm just asking you a question, how many of you like ice cream? Please. I like ice cream, especially my daughter loves mostly. Right, okay, great. Can you just tell me how much amount of microbes may be present in the ice cream? Anybody? Please. I know you are very starving, I know, but I will complete it shortly. Yes. Anybody? Okay. This is the result. See the first one, 9. Like 1 gram of ice cream may contain 2 lakh microorganisms. This is the FSSAI standard. Here you can see. First one. 2 lakh CFU per gram may be present in your sample. Right. This is what I am trying to say. The total plate count is entirely different than the E. coli and the total coliform. Here you can see the total coliform. If you are taking a 50 gram or 50, right, 50 total coliform present in the 1 gram of sample. But the sample should not contain any E. coli. As per the FSSAI, 9 parameters you mandatorily do it. Right. 4 years back, Kerala faces the very big flood. That time the Kerala government announced a red alert on listeriosis. Listeria, it is a zoonotic infection. All the flood water will be mixed with toilet water, septic tank waste and all the household waste. So people are highly affected by listeria. So they have announced the listeria red alert. There is a chances if the waste water or the waste will be mixed with your drinking water as well as the food. So this is mandatory. We have to do some of the analysis, right? If it is a fish sample, it is a seafood sample, you have to do mandatorily, you have to do Vibrio cholerae and Vibrio parahemolyticus. Whenever you are taking this kind of meat, you have to take a 25 gram of sample, not a 1 gram, not a 1 ml. Even you can dilute or homogenize the sample, you must take only 25 gram of sample. That is a mandate. Okay. And as per FSSAI, right, even if you are using any kind of spices, right, that should not contain any kind of organism. Here I am listed out some of the 9 important parameters. Aerobic spore count and anaerobic spore count. Clostridium, aerobic spore count is bacillus. These 2 parameters you mandatorily you have to analyze, then only you can market it. You may ask me like, sir, everything they have analyzed, sir. Why you may ask about maybe the sample may be contaminated. Only one thing, the biggest companies, every batch they are doing the analysis. But the smallest companies, for getting the license, they have done the analysis. After that, they didn't follow that. Each and every batch production, they must do it. Whenever you are placing in the industry, just insist the person who are in charge to you, you have to mandatorily test the sample. Otherwise, we have to give poison to our fellow members. Right. And Codex Alimentarius, it is an important one. It is recognized by all the international standards. They said it is a risk assessment of ABR, and they mentioned what are all the criteria we have to follow to control it. I will share this PPT with you just to go through it. Okay. And this is an important one. How to identify it, how to do the monitoring and surveillance, and how to control it. They have provided 16 principles for control it for each and every human being, as well as they are providing the control measures and what the competent authority has to do. The legislation department or the country fellows, what the politician, the country leader has to do. All the informations they have given. So before we have to do all this kind of work or all this kind of research, or whenever you are receiving the sample for this one, you just follow it. It is related to food. You have to follow FSSAI as well as Codex to mitigate all these problems. Okay. So that's all about the presentation. I think I keep my promise. Complete in 15 minutes.
Now the session is open for queries.
Thank you. If I know the answer, I will tell you. Otherwise, definitely I will refer it and tell you. Please, if you have any doubt, you can ask me. Thank you for patiently listening. Time is almost 1:20. Yeah. Thank you. Please, any doubt, you can ask me.
Sir, good afternoon, sir. The session was interesting. I have a general doubt, sir. The canned food containing most of the சார் கேண்ட் ஃபுட்டில் நிறையா கண்டாமினன்ஸ் இருக்குன்னு சொல்கிறாங்க சார். நம்ம மேனுஃபேக்சர் பண்ணுறப்போ நம்ம ஜிஎப்பி ஜிஎல்பி எல்லாம் ஃபாலோ பண்ணி தானே பண்ணுறோம். தென் எப்படி சார் கேண்ட் ஃபுட்டில் கண்டாமினன்ஸ் இருக்காது, என்ன பாசிபிலிட்டிஸ்?
சூப்பர் கொஷின்மா, தேங்க்யூ. நார்மலி ஷெல்ஃப் லைஃப் ஸ்டடீஸ் நம்ம ஒன்று பண்ணுவோம். ஒரு ஃபுட்டை பேக் பண்ணதுக்கப்புறம் 6 monthsன்னு கொடுத்துருப்பாங்க, 9 months கொடுத்துருப்பாங்க, சில ஃபுட்டுக்கு 3 monthsன்னு கொடுத்துருப்பாங்க. ஓகே, அந்த கண்டிஷன் கம்ப்ளீட்டாக நம்ம டெஸ்ட் பண்ணுவோம். ஓகே, அந்த டெஸ்டிங்கில் சாம்பிள் சம்டைம்ஸ் ஃபெயில் ஆகும். அந்த ஃபெயிலியரை நோட்டீஸ் பண்ண மாட்டாங்க. இது லோக்கல் வெண்டாஸ். பெரிய லெவல் வெண்டாஸ் என்ன பண்ணுவாங்க அப்படின்னா, nowadays people are using a different kind of can material. ஓகே, அந்த கேன் மெட்டீரியல் நீங்கள் கேன் ப்ராசஸ் பண்ணதுக்கப்புறம் அதை வந்து ஹாட் வாட்டரில் எக்ஸ்போஸ் பண்ணுவாங்க சம் டைம். அண்ட் இல்லைன்னா இரிடேஷன் மெத்தட் போவாங்க. அந்த இரிடேஷன் மெத்தட்லேயோ இல்லை ஹாட் வாட்டர் எக்ஸ்போஷர் மெத்தட்லேயோ ஏதாவது ப்ராப்ளம் இருந்துச்சுன்னா டெஃபினட்டாக கண்டாமினேஷனுக்கான பாசிபிலிட்டிஸ் உண்டு. ஓகே, அண்ட் இன்பில்ட் ஃபார் எக்ஸாம்பிள் ஃபிஷ்ஷஸ்லாம், இப்போ மைக்ரல் ஃபிஷ்ஷஸ்லாம் இருக்குது, மேக்ரல் ஃபிஷ்ஷஸ்லாம் பாயில் பண்ணிவிட்டு தென் தே வில் soak it in the brine water. The brine water level, PPT level is around 3 to 5. 3 to 5க்கு மேலே brine water solution போடுச்சுன்னா ரப்சர் பண்ணிவிடும் உங்கள் டிஷ்யூ எல்லாம், சால்ட் வாட்டர் ரப்சர் பண்ணிவிடும். ஓகே, ஸோ அதனால் அதுக்கு மேலே யூஸ் பண்ண மாட்டாங்க. ஸோ ஹாலோஃபிலிக் ஆர்கானிசம் may survive in that. So we have to tested everything, what kind of material we are adding. So mostly canned food லையுமே benzoate அதிகமாக யூஸ் பண்ணுவாங்க. Okay, if you are claiming that is a no preservative, there is a chances for contamination. You have to process it properly. The final stage process is very important. Even the microbiological role is very crucial in all the food system, right? Each and every stage they will analyze they will do the testing, right? Even the shelf life studies la 3 months உங்களுக்கு shelf life நல்லா இருக்குன்னா they will give only 2 months. ஒரு சேஃபர் சைடுக்கு தான் கொடுப்பாங்க. ஸோ இஃப் தே கிளைம் தட் இஸ் a 9 month the product will be good. Suppose the product actually it will be good for 12 months actually. 10 monthsக்கு நல்லா இருக்கும், அவங்க ஆனால் 9 months தான் கொடுப்பாங்க. ஓகே, ஸோ ஷெல்ஃப் லைஃப்பில் இந்த மாதிரி இஷ்யூஸ் வரது ரீசன் ப்ராப்பராக அங்கே வந்து ட்ரீட்மெண்ட் கொடுத்துருக்க மாட்டாங்க. ஹாட் வாட்டர் எக்ஸ்போஷரோ இல்லை இரிடேஷனோ ப்ராப்பராக அங்கே பண்ணியிருக்க மாட்டாங்க. தட் இஸ் த மெயின் ரீசன். ஓகே, தேங்க்ஸ்.
சார், another doubt சார், இப்போ டைரி இண்டஸ்ட்ரீஸ்லையும் மேக்ஸிமம் இந்த மாதிரி கண்டாமினேஷன் சீக்கிரம் கெட்டு போயிருதுன்னு சொல்கிறாங்க சார். அதை ப்ரிவென்ட் பண்ணுறதுக்கு ஏதாச்சும் ஐடியாஸ் இருக்கா சார்?
ஒன்லி திங் ப்ராசஸிங் தான். we have to use some amount of preservatives, that is mandatory. Because once upon a time when I was young, I still remember my village is covered with full of agricultural land. Now it is completely covered with the concrete blocks, right? So we have to feed this much people. Population ரொம்ப increase ஆயிருக்கு, and one side we are reducing everything. So we totally depend on the processed food. இப்போ processed foodங்கிறப்ப நம்ம preservative ஆட் பண்ணி தான் ஆகணும். as per FSSAI guidelines நீங்கள் யூஸ் பண்ணலாம். யூஸ் பண்ணோம் அப்படின்னா நம்மளால் அவாய்ட் பண்ண முடியும், கண்டாமினேஷன் அவாய்ட் பண்ண முடியும். இல்லை அப்படின்னா ப்ராப்பர் ஸ்டோரேஜ் 4 degree to 60 degree we call it as a danger zone. Your refrigerator temperature is 38 degree Fahrenheit, approximately 4 to 7 தான். எக்ஸாக்ட் 4 எல்லாம் கிடையாது, 4 to 7 வரைக்கும் இருக்கும். Better you can keep it below 4 degree Celsius, or else you have to cook the food for more than 60 degree, then you can store it in the refrigerator for some extent. இதுதான் ஆப்ஷன். ஒன்று, you have to go with the food, you have to do some temperature related work, store it in the cold or heat it, or you can use some preservatives. That is the only way to extend the shelf life. Thank you.
Thank you, sir.
Thank you.
Any other queries?
Good afternoon.
Good afternoon.
Good afternoon, sir. Thank you for the session, sir. Sir, food materials எல்லாமே வந்து மைக்ரோப் கண்டென்ட் இருக்கும் அப்படின்னு சொல்லி எக்ஸாம்பிளுக்கு வந்து 1 gram of ice creamல வந்து 2 lakh of microbes content இருக்கும் அப்படின்னு. ஸோ அந்த லோ டெம்பரேச்சர் ரெஃப்ரிஜரேஷன் ஸ்டோர் பண்ணியும் அவ்வளோ மைக்ரோப்ஸ் வந்து எப்படி அந்த கண்டிஷன்ல சர்வைவ் ஆகும்?
ஸ்டார்விங்கில் எஃப்எஸ்எஸ்சி ஸ்டாண்டர்ட் எப்போதுமே என்ன சொல்கிறாங்க அப்படின்னா அவ்வளோ இருக்குங்கிறத சொல்லலை, அவ்வளோ வரைக்கும் அவங்க அக்செப்டன்ஸ் லெவல் கொடுத்துருக்காங்க. டிபிசி அப்படிங்கிறது டோட்டல் பிளேட் கவுண்ட் ஆர் டோட்டல் ஏரோபிக் கவுண்ட் ஆர் டோட்டல் வயபிள் கவுண்ட்டுங்கிறது நார்மல் ஆர்கானிசம், ஒரு கமென்சல் ஆர்கானிசமாக இருக்கலாம், அது எல்ஏபியாக இருக்கலாம், இல்லை பேத்தோஜெனிக் ஆர்கானிசமாகவே கூட இருக்கலாம். பட் மேக்ஸிமம் 2 lakh வரைக்கும் இருக்கலாம்னு சொல்கிறாங்க. அது இருக்குங்கிறது அர்த்தம் கிடையாது. பட் தேர் இஸ் அ சான்சஸ். அண்ட் ஆல்ரெடி நான் சொன்ன மாதிரி தான் நீங்கள் கோல்டு ஸ்டோரேஜில் பண்ணப்போ சம் ஆஃப் த ஆர்கானிசம் வில் கோ டு த ஸ்டார்விங் கண்டிஷன், ஸ்டார்வேஷனுக்கு போயிடும். சில ஆர்கானிசம் ஆக்டிவாக இருக்கும், சம் ஸ்போர்ஸ் கேன் பி ஆக்டிவ், ரைட்? அண்ட் செகண்ட், when I have taken the class, I told you 3 criteria. One is how much you are intake, எவ்வளோ ஆர்கானிசம் உள்ளுக்கு போகுது, இன் விச் how many organisms are pathogen. டோட்டல் பிளேட் கவுண்டை நம்ம பேத்தோஜன் லிஸ்ட்டில் கொண்டுக்க மாட்டோம். ஸோ அதனால தான் நம்ம ரிமைனிங் பேராமீட்டர்ஸ் பண்ணுறோம். 3 parameters mandatorily we have to do for all the food sample. மார்க்கெட்டில் கிடைக்கிற எந்த பேக்டு ஐட்டம் எல்லாமே 3 டெஸ்ட் இல்லாமல் வெளியே வராது, ஆஸ் பர் ஃபுட் நான் சொல்கிறது. ஒன் இஸ் டோட்டல் பிளேட் கவுண்ட், செகண்ட் ஒன் இஸ் டோட்டல் ஈஸ்ட் அண்ட் மோல்டு கவுண்ட், தேர்டு ஈகோலை, டோட்டல் கோலிஃபார்ம் அண்ட் ஈகோலை பார்ப்பாங்க. ஸோ ஈகோலை என்ட்ரிக் ஆர்கானிசம் இருந்தாலே உங்களுக்கு அந்த சாம்பிள் ரிஜெக்டட். இது இல்லாமல் சாம்பிள் ஸ்பெசிஃபிக் சில பேராமீட்டர்ஸ் பார்ப்பாங்க, ஸ்டாஃப் ஆரியஸ் பார்ப்பாங்க, லிஸ்டீரியா பார்ப்பாங்க, கம்ஃபிலோபாக்டர் செக் பண்ணுவாங்க. எஸ்பெஷலி மோஸ்ட்லி நம்மளுடைய சாம்பிள் எல்லாமே எக்ஸ்போர்ட்டில் ஃபெயிலியர் ஆகிறது ஒன்லி ஒன் ரீசன், சால்மோனல்லா. If you are effectively identify the salmonella, if you can easily identify the salmonella, you will become a billionaire in abroad. If you are very strong in the identification of Shigella and Salmonella, right? So இந்த parameters எல்லாமே பார்ப்பாங்க. இது எல்லாமே ஜீரோன்னா தான் ஈவன் 2 lakhs இருந்தாலும் அலோவ் பண்ணுவாங்க, பிகாஸ் that does not contain any pathogen. அதனால தாராளமாக எடுத்துக்கலாம், no issues.
Thank you, sir.
Thank you.
Good afternoon, sir.
Good afternoon.
I have a doubt. 1 gram of ice cream வந்து 2 lakh microbe வந்து இருக்கும் அப்படிங்கிற ஒரு அக்செப்டன்ஸ் லெவல், அந்த அளவுக்கு லிமிட் இருக்குன்னு சொல்றீங்க. ஸோ ப்ரோபயோட்டிக் ஐஸ்கிரீம் எடுத்துக்கிறப்ப அதுக்கு ஏதாவது அக்செப்டன்ஸ் லெவல் இருக்கா?
எல்ஏபிக்கு அந்த மாதிரிலாம் இல்லை. இப்போ ரீசென்ட்லி ஒன் கம்பெனி தே லான்ச்டு ப்ராடக்ட் லைக் அ ப்ரோபயோட்டிக் கர்ட் itself is a probiotic. அது ப்ரோபயோட்டிக்கிற டெர்மினாலஜி தேவையில்லை. பட் தே ஹவ் ஆடட் 2 more கல்ச்சர் இன்டு தட். ஓகே, நீங்கள் எல்ஏபி எவ்வளோ வேணாலும் எடுத்துக்கலாம், அதனால உங்களுக்கு ப்ராப்ளம் இல்லை, ரைட்? தாராளமாக எடுத்துக்கலாம். எல்ஏபி அதில் இருந்துச்சு அப்படின்னா, சில ப்ராடக்டில் எல்ஏபி இருக்கக்கூடாது. இருந்துச்சுன்னா இட் வில் ஆல்சோ ப்ரொடியூஸ் அ பயோஃபிலம், இட் வில் செக்ரேட் த பயோஃபிலம், தட் there is a chances for just contaminate the product. Contaminate in the sense bad odor வரலாம். ஓகே, எல்லா இடத்துலையும் எல்ஏபி பெனிஃபிஷியல்னு சொல்ல முடியாது எல்லா ப்ராடக்ட்லையும். பட் சம் ஆஃப் த ப்ராடக்ட் இட் இஸ் குட், இருக்கலாம், தப்பே இல்லை, எவ்வளோ இருந்தாலும் ப்ராப்ளம் இல்லை.
அண்ட் லாக்டோஸ் இன்டாலரண்ட்டாக இருக்காங்க இல்லையா, ஸோ அப்படி அவங்களுக்கு இருக்குங்கிறப்ப அவங்களுக்குன்னு ஒரு அக்செப்டன்ஸ் லெவல் கொடுத்து தான் ஆகணும் ப்ரோபயோட்டிக்காக இருந்தாலும்.
இல்லை, ப்ரோபயோட்டிக் இந்த சென்ஸ் லாக்டோஸ்க்கு தான் உங்களுக்கு இன்டாலரன்ஸ், ஆர்கானிசம் லெவல் அது பெருசாக அஃபெக்ட் பண்ணாது. ஈவன் யூ கேன் டேக் லைக் அ நீராகாரம். In China, it is a very commercial biggest market. நீராகாரத்துக்கு fermented foodக்கு it is a very value, right? And they are sell it in the market. Okay, அது நீங்கள் அந்த மாதிரி எடுத்துக்கலாம், அதில் பெரிய உங்களுக்கு டாலரன்ஸ் அஃபெக்ட் ஆகாது ஒன்றும். இன்னொன்று மார்க்கெட்டில் இப்போ அவைலபிளாகவும் இருக்குது, இன்டாலரன்ஸ் யோகர்ட்லாம் அவைலபிள். ஸோ if you find out one of my student Anjali, she is now working in NIFTM, she is doing her PhD in NIFTM Kunli. She was a lactose tolerant. She have developed 18 yogurt product along with the team members. She is a lactose tolerant yogurt, less lactose yogurt we have prepared, added some amount of our indigenous product into that. We are going for submitting the paper அந்த பிறகு நான் ஷேர் பண்ணுறேன், you can refer it for that, for those people, lactose intolerance people.
Thank you, sir.
Thank you.
Any other queries? Thank you, sir.
Thank you.
I now request Dr. B. Ramalingam, Scientist Semi-Deputy Director, to present a token of love to our keynote speaker. Thank you, sir. A thankful heart is not only the greatest virtue, but the parent of all the virtues. I would like to invite Ms. R. Gautami from 2nd MSc Microbiology to deliver the vote of thanks.
இங்கே சொல்றதா
ஒன்ஸ் கொஞ்சம் ஃப்ரெண்ட்ஸ்க்கு டிஸ்கஸ் பண்ணனா வில் கெட் சம்திங். ஏதாவது ஒரு லிங்க் இருக்குது யாருக்காவது நம்ம ஃப்ரெண்ட்ஸ்க்கு
It's my honor to extend a heartfelt vote of thanks. I would like to express our sincere gratitude to our esteemed chairperson, Dr. M. Pungodai, for her unwavering support and contribution to this session. A sincere thanks to our distinguished guest, Dr. S. Vignesh, for his insightful session on food pathobiome and food resistance, the big future. Your session has greatly enriched our understanding of the complex relationship between food pathogens and antibiotic resistance. Thank you, sir, for sharing your valuable knowledge with us today. Once again, thank you all.
Now it's time for lunch break. Lunch will be provided in Arts Canteen behind Pavayarangam. After having your lunch, kindly be here back by 2 p.m. sharply. I repeat, after having your lunch, kindly be here back by 2 p.m. sharply.
Students, please, if you assemble back exactly by 2 p.m., we will have the valediction program where that will be completed as soon as possible, whereby by time you can go home. Very simple. Thank you.
Students are requested to assemble quickly. The program is about to start.
Students, maintain silence. Silence. Second UG, silence. Department of Microbiology, Vinod Kumar.
afternoon to everyone present here. We welcome you all to the valedictory session of 18th Louis Pasteur National Symposium.
A warm welcome paves the way for meaningful exchanges. To extend such a welcome, I invite Mr. Bharat TT to deliver the welcome address.
Good afternoon, everyone gathered here. It's a privilege to stand before you as we gather for the valedictory ceremony of 18th Louis Pasteur National Symposium. Over the past 2 days, we have witnessed enriching discussions, insightful presentations, and an incredible exchange of knowledge on alternative medicine for combating infectious diseases, AMCID 2025. We are honored to have with us Dr. B. Ramalingam, whose work continues to inspire researchers and professionals alike. We welcome you, sir. Next, it's our pleasure to welcome our guiding force behind our department, Dr. S.S. Sudha, DBT Star Overall Coordinator. We welcome you, ma'am. A special welcome to the organizing committee of 18th Louis Pasteur National Symposium, whose dedication and hard work have ensured the seamless execution of the symposium. We are delighted to welcome our head and faculty members from our department as well as from the other department and institutions who have joined us here today. Your presence truly enriches this gathering. And a warm welcome to our enthusiastic student participants, both from our college and from other institutions. Your engagement and curiosity have made this symposium meaningful, and we appreciate your active involvement. With that, let us proceed with the valedictory session celebrating the success of 18th Louis Pasteur National Symposium. Once again, a warm welcome to one and all. Thank you.
Thank you, Bharat. As we come to the conclusion of the symposium, it is time to reflect on the insights gained and the knowledge shared. I now request our esteemed guest speaker, Dr. B. Ramalingam, to deliver the valedictory address.
Yeah, so good afternoon again, and it is very privilege and honor to be here around the young minds and giving me an opportunity to be here around you. So for all beginnings there will be an end, and beginning is very important, but the end should not be always end. It has to be continued and it has to be renounced. So in this manner, like I understand the management and the staff of the department has made a tremendous effort. And I should say you people are really privileged to have these kind of eye-openings or conferences being listened by you at an early career stage. So I hope you are BSc and MSc here, and you are being given exposure to all the research avenues, and these things can give you a source of inspiration where you can understand the role of microbiologist and what are all the areas they can explore in terms of research or in terms of employment or in terms of empowerment. So all these exposure, awareness, everything comes only through these kind of conferences. And I should thank the management and the department and Madam that they have been successfully carrying out this endowment lecture or conference in the name of Louis Pasteur. It is Louis Pasteur, okay? So it is not Louis Pasteur. You should say Louis Pasteur because of the French term, the last syllable doesn't always come. So you should say Louis Pasteur. And it is a very appropriate name, and I don't know how many of you know what Louis Pasteur has identified. Any idea like what is his contribution on Louis Pasteur in the terms of microbiology? 1%. Vaccines. Other than that, pasteurization. Okay, other than that, any organism, can you think? He has discovered multiple roles, okay, and yeast was the major discovery. Other than that, fermentation product, pasteurization, all those things were his contribution. So the French government appreciated, and then they had the Institute of Louis Pasteur, and then this has been spread across. And there is one institute in Ooty also. So I hope you people would have gone there to visit that also. So that is the contribution of microbiologist, and people should always create an interest, because you are an expert in the field now. After baccalaureate, after masters, you are recognized as a microbiologist. So in that field, you should always be aware of what is happening across the country or in your field or in your knowledge. And then you need to imbibe and you expose yourself to the society as a microbiologist. Your father, mother will see you as a daughter or son, but you should remind them, I am a microbiologist. When you say that, you ask them repeated hand washing, allowing to make sure contamination of foods. You keep them aware so that you are a microbiologist. So it is always the microbiologists are considered as the OCDs, always they are very spick and span about washing their hands, making cleanliness and all those things. So your microbiology knowledge should not restrict to that part. You should always explore it, see the day-to-day activities, what is the contributions of microbe in our day-to-day life. Now it is said that the gut is the second brain. So as a good organ as brain, the gut microbiome is directly linked to the brain activity. So there are humpty number of opportunities to you to success in this field. So you please make sure for that you need to excel in this field. So try to explore the opportunities given by your staff, given by the college, make use of it and excel in your field. So with this small advice, I thank the management and the staff for giving me this opportunity, and I'll be happy to be here for any support if I can. Thank you very much.
Thank you, sir. We now request Dr. S.S. Sudha, DBD Star Overall Coordinator, to felicitate the gathering.
Good afternoon all. I am very happy and proud to be part of this 18th Pasture Symposium. As rightly pointed out by our guest, Dr. Ramalingam sir, in the valedictory address, in what extent as a student or as a society person you can contribute to your neighborhood as well as your family, you have to think and work accordingly. And these 2-day deliberations with 6 sessions, one is by online, and all 5 sessions, it's a eye-opener for you, as well as you might have received plenty of information from the sessions. Hope you all are inspired. And sir told that few of the students, they are working in Mycobacterium tuberculosis and they wanted to visit their institute in Chennai. Sir agreed for that also. Thank you, sir, at this juncture. And the purpose of making this type of conferences symposium is to bring the resource persons from all over the country. It is not possible for all the students to go and attend the conference in different places. So it is possible to bring the persons to our campus so that every student can be given the opportunity to meet the resource person, so that what extent you can get some internships that you can make available with the help of the resource person. So whoever is coming to our campus, they are helping hand to our students. They can support, and yesterday also Dr. Gandhi Rajan told that they will give preference for microbiology students of Dr.N.G.P for placement. Even though he is working in some other institution, some other hospital, in Royal Care Hospital, he is ready to give opportunity for microbiology students of NGP. So in that way, all resource persons are giving helping hands for our students. It's opportunity for you to make use of the facilities available in our campus and the opportunities made by the department. I thank at this juncture the department head and all faculty members and the organizing secretaries sitting on the chair, dais, Dr. Sasikala, Dr. A.M. Ramachandran and Dr. Mahendran, under the leadership of Dr. R. Rangaramanujam and all other faculty members who have given lot many work for this symposium, and I have been observing for the past 1 month, and they are working hard for this, and we could be able to bring the resource persons, that is, delegates from other colleges too, and you all have come out with good abstract, and we are going for proceedings with that few papers also. So wish you all good luck. Thank you for the opportunity.
Thank you, ma'am, for your valuable words of wisdom. Recognition is the greatest motivator, and it's time to honor the best presentations of the symposium. To announce the prize winners for the oral and poster presentations, I invite Bharat Tete from 3rd UG Microbiology.
Good afternoon, everyone. We have now reached the most exciting part of the symposium, the prize distribution for the oral and poster presentations. Let's begin with the oral presentation. 3rd prize, Ashwati from RVS College. She is an online presenter, so she is not available now. 2nd prize goes to Ashok SJ and from Sri Ramakrishna College of Arts and Science. First prize goes to R. Monish Kumar from Dr.N.G.P Arts and Science College. Moving on to the poster presentations, other college category, 3rd prize goes to Moving on to the poster presentations, other colleges category, 3rd prize goes to M. Srimugi from Gandhigram University. Second prize goes to V. Gautami S. Akshaya from Sri Venkateshwara Arts and Science College. Any one student from Sri Venkateshwara Arts and Science College, come and collect your certificates. First prize goes to R.K. Abhinaya Varna M. Neshiha from Hindustan College of Arts and Science. Next, the poster presentation prize winners from our college category. 3rd prize goes to S. Yuvashri from 3rd BSc Microbiology. Second prize goes to M. Shweta J.K. Yashida from first BSc Microbiology. And finally, the first prize goes to J. Aroma Jesu and Sanjana Rajesh from first MSc Microbiology. I request the dignitaries to present the certificates to the participants from other departments and colleges. As follows, any one student from our college biotechnology department come to the dais and collect your certificate. Any one student from biotechnology department, please come to the dais and collect your certificate. Any one student from each college can come to the dais and get all of your certificates from the dignitaries as follows. Sri Venkateshwara Arts and Science College, Bharatidasan College of Arts and Science, Sri Ramakrishna College of Arts and Science, any one student from Hindustan College of Arts and Science. Now certificates will be awarded to the judges for oral presentations and judges for poster presentations as well as the session chairs for all sessions. Ms. C.R. Aarti from Department of Biotechnology. Dr. S. Saranya from Department of Biotechnology. And Dr. M. Pungodai from Department of Biotechnology. Thank you, dignitaries. A big congratulations to all the prize winners. Your dedication and hard work have been truly remarkable. I also want to take a moment to appreciate each and every participant who presented their paper and poster. Your efforts and insights and passion for knowledge have made this symposium. Winning is important, but the courage to present and share ideas is equally valuable. Keep striving, keep learning, and keep inspiring. I sincerely thank our esteemed dignitaries for graciously presenting the prizes to the winners. Once again, I thank you all.
Thank you, Bharat. As we conclude AMCID 2025, we would like to hear your thoughts on this symposium. Your feedback will help us improve and make future editions even more enriching. We welcome participants to share their experience and insights.
As the saying goes, gratitude turns what we have into enough. To extend our appreciation, I invite Ms. Ridhanya VK from 3rd BSc Microbiology to deliver the vote of thanks.
Gratitude is a powerful catalyst for happiness. It is a spark that lights a joy in your soul. I deem it a great honor and privilege to propose the vote of thanks on this 18th Louis Pasteur National Symposium on Alternative Medicine for Combating Infectious Diseases, AMCID 2025, organized by Department of Microbiology under DBT status scheme. It is a distinct privilege to prize the keynote speakers of AMCID 2025, Dr. N. Thammaselvam, Dr. Gandhi Rajan, Dr. Natesan Manikam, Dr. G. Rajmohan, Dr. B. Ramalingam, and Dr. S. Vignesh from various university and research platforms for contributing us with their field of expertise. Thank you all. A special thanks to our beloved judges, Dr. K. Arun Gandhi, Professor, Dr. M. N. Kadaravan, Professor, Dr. M. Shanmugh Vadivu, Professor, Dr. Radha Palaniswami, Associate Professor, Dr. S. Saranya, Assistant Professor, and Ms. C.R. Aarti from Department of Biotechnology of our college. Your time and dedication made us to reward the student participant. Thank you all. We are grateful to our college management, founder and chairman, Dr.Nalla G. Palaniswami, and our esteemed Madam Secretary, Dr. Thavamani D. Palaniswami, for inspiring us to work with dedication. It's our privilege to thank Dr. P.R. Muthuswamy, Director Academics, Dr.N.G.P Group of Educational Institution, for his inspiring mentorship. We convey our profound gratitude to Dr. K. Ramamurthy, Director Academics of Dr.N.G.P Arts and Science College, for his support, vision and motivation. With gratitude, we thank Dr. S. Saravanan, Principal, Dr.N.G.P Arts and Science College, for being a driving force behind our achievements. Our pleasant thanks to all the deans of various portfolios for being a great support in all our efforts. Our heartfelt thanks to Dr. S.S. Sudha, DBT Star College Overall Coordinator, for motivating us to strive for the best. Thank you, ma'am. We would like to thank Dr. J. Rangaramanujam, Head Department of Microbiology, for his guidance and strategic direction for the success of the symposium. Thank you, sir. A big thanks to the organizing secretaries of this 2 days National Symposium, AMCID 2025, Dr. A.M. Ramachandran, Associate Professor, Dr. C. Sasikala, Professor, and Dr. R. Mahendran, Assistant Professor, for the meticulous planning to make the symposium a grand success. Thank you all. Our heartfelt thanks to Dr. N. Vidya, Professor, Dr. S. Sendil Prabhu, Professor, Dr. S. Karthik Sundaram, Associate Professor, Dr. J. Devakumar, Assistant Professor, and Ms. N. Nivedita, Assistant Professor, for their fostering contribution to the symposium. Thank you all. A deepest gratitude to other departments, faculty members, non-teaching staff members who had been contributed a lot to make this symposium a resounding success. Thank you all. We mention our deep sense of appreciation to all the research scholars and student delegates of our institution and other institutions. Thank you all for an enthusiastic participation. A special appreciation to MOC team for guiding us throughout the event and volunteers for their seamless work for the symposium. Thank you all. Finally, it essentially to thank all our staffs, technical staffs and housekeeping staffs to make this forum shines better. Thank you all. We wanted to take a moment to thank our national sponsoring agency DBT Government of India and Purani Hospital Suppliers Private Limited, who played a key role in the success of National Level Symposium AMCID 2025. Once again, we thank you all for your cordial cooperation. Thank you.
Thank you, Radhanya, for your words of gratitude. Let us all now rise for the national anthem. Students, kindly stay back in the hall as we have photo sessions. First UG students come forward to take photo. To take group photo.
Ready for the photo session?
Second UG students, get ready for the photo session. Second UG students, come forward. Followed by second UG, third UG, get ready. Second UG students, be ready. Second UG, get ready. Come forward. Gayatri Akka, please come forward. students, you're next. Please be ready.
Followed by third UG, first PG, get ready.
Followed by first.
First PG, come forward. First PG students, come forward. Followed by first PG, second PG are requested to come forward. Research scholars are requested to come forward. Research scholars are requested to come forward. Second PG, come forward. for MOC are requested to come for a photo session. Next, scholars are requested to assemble in the MOC students are requested to assemble on the desk.